Welcome to the Feed the Future Innovation Lab for Legume Systems Research Annual Virtual Convening, Public Forum Day 2. Thank you for joining us. We look forward to continuing our conversation on innovative approaches and discussions on legume systems research. My name is Doreen Gordon, Fiscal Officer for the lab. I will be facilitating our session today. A few details on our meeting format. You can view this session in the Hoover platform or on the Zoom platform. In today's session, all presentations with the exception of one are in English. If you need translation to French or audio translation from French to English, look for the globe icon in the bottom menu in either Zoom or Hoover. After selecting the globe icon, you can select your preferred language. If you continue to hear an additional language, try selecting mute original audio. Also, if you have any technical difficulties joining the Hoover platform, please select Join in Zoom. If you do join in Zoom, please also keep this session open and available in the Hoover platform. This will allow you to participate in Q&A, chat, polls, community, or to access the session agenda, as all of these features are available in Hoover exclusively. I would also like to highlight the poster gallery in Hoover, featuring research posters presented by students working with the lab projects. After today's sessions end, we encourage you to visit the poster gallery and select Visit Booth of the posters you wish to view. When visiting the booths, make sure you scroll down to the handouts where you can download a PDF, the poster. I would also like to highlight the community feature. Here you will find additional resources on many topics and be able to engage in ongoing conversations throughout the convening. Please note the chat feature in Zoom is limited during this session. Please use the chat in Hoover. Yesterday, we asked you to share your favorite way to eat legumes or your favorite legume recipe. I'd like to share some results of that poll. Cowpea was noted as the most favorite legume. Others mentioned include pigeon pea, chickpeas, mung beans, and red common beans. Beans and rice and akara were noted as the most popular ways to enjoy legumes. Other dishes included bean stew, chickpea curry, bean tacos, baked beans, and boiled beans. This all shows the incredible versatility of legumes. Packed with protein, no matter how you enjoy them, legumes are always a great nutritional choice. Thanks to everyone who participated in yesterday's poll. At this time, Dr. Barry Pittenry, Legume Systems Innovation Lab Director, will share a few welcoming words and announce the winners of our student poster competition.
Thanks, Doreen. I'd like to welcome everyone to day 2 of the Legume Systems Innovation Lab Annual Global Convening Public Forum. Yesterday, we heard updates and highlights of activities from 5 of our Legume Lab research projects, and today we will continue with the research session. As I mentioned yesterday, building and investing in the next generation of legume researchers is an important commitment of the lab. I especially enjoyed yesterday's student presentations and look forward to hearing from more students during today's research session. Currently, our projects are supported by 38 students, including 22 women, as they pursue degree programs globally. I really hope you all have had an opportunity to view the research posters submitted by the outstanding students supported by Innovation Lab projects. As a reminder, they can be viewed by clicking the Posters tab in the WOVA platform. It is also a great pleasure to announce the winners of the student poster competition. We will award 2 students top honors and the opportunity to participate in the Legume Systems Innovation Lab Grant Writers Workshop, where they will learn effective strategies on communicating their science. The 2-week workshop will be led by international science communication expert Mark Beyer of Beyer Strategic Concepts and offers individuals expert feedback on how to write winning proposals and engaging narratives to advance their research priorities. I'm happy to announce the Feed the Future Innovation Lab for Legume Systems Research Poster Competition for 2022. Winners are Juno Bruno Nyade and Ya Nesamo. Juno Bruno Nyadi is pursuing a master's degree at University of Cheikh Anta Diop, UCAD of Dakar, Senegal. His poster is entitled Improving Food Security and Nutrition Through Dissemination of Dual-Purpose Cowpea Varieties into Farming Systems in Senegal. He's supported by the Dual-Purpose Cowpea Project led by Augustine Ubar at Kansas State University, and in Senegal by Eloy Faye at Isra Saras. Ya Nasumo is pursuing a master's degree in the Department of Crop Sciences and Soil Science at Kwame Nkrumah University of Science and Technology, KNUST, in Kusami, Ghana. His poster is titled Interaction Effect of Genotype and Environment on Yield and Yield Components of Cowpea in the Dry Savannas of Ghana. He is supported by the Improved Cowpea Varieties for West Africa project led by Phil Roberts at University of California, Riverside, and in Ghana by Francis Kusi at CSIR Sari. Congratulations to Junior and Yawa to all the students and to all the students who submitted a poster for this convening. You are our future and we expect big things from each of you. Best of luck in your studies. And thank you to the TMAC members that served as judges for the poster competition. At this time, we would like to jump into today's research session. So yesterday, 5 of our projects, and we will jump into the research session currently, which I'll moderate. Yesterday, 5 of our projects provided updates and continued, and today we will hear from our remaining projects. As we mentioned yesterday, including along with the project research updates, we will also feature presentations by students supported through the lab. We're excited to have these students showcase their work for you today. The format of the research session will be a pre-recorded presentation by the project team along with any student presentations related to the presenting project. These pre-recorded presentations will be followed by a short live question and answer period. To submit a question, please use the Q&A feature in the right panel of your convening page on Wova during the presentation. Since our Q&A time is very limited, we ask that all participants monitor the Q&A and vote up questions that are of the most interest to you by clicking the thumbs up icon next to the individual question. In the open Q&A session, at the end of each presentation, we'll be selecting the question with the most upvotes for further elaboration. Also, to help us in managing your questions, please preface your question with the numeric designation for the presentation. I'll provide the corresponding numeric designations before each presentation. Our first presentation is Genetic Improvement of Dry Beans for Bruchid Resistance for Southern Africa. This project is ongoing and led by Dr. Juan Osorno of North Dakota State University. Immediately following their presentation will be a presentation given by 2 students, Maria Mazala and Madrine Chinzi, studying under Dr. Osorno's project. Maria is a master's candidate at North Dakota State University, and Madrine is a master's candidate at University of Zambia. Both individuals are from Zambia. As I mentioned before, the Q&A designation from Dr. Osorno's presentation is 6 and 6A for the student presentation. Presenting today from Dr. Juan Osorno's team is Dr. Kevin Kamfwa. The Q&A designation for this project is 6.
Good afternoon, ladies and gentlemen, or good morning, depending on where you are. My name is Kelvin Kamfo. I'm the co-PI for the project entitled Genetic Improvement of Dry Beans for Bruchid Resistance for Southern Africa. And I'll be presenting on behalf of quite a large group that we have working on this project. As part of the introduction, the focus of the project is on the common bean weevil, which is Acanthocylus obstetricus, It's the most prevalent type of weevil that we have in Southern Africa, and it's responsible for major post-harvest losses, and it also leads to the loss of quality of the seed and also the germination ability of the seed. So it's quite a devastating pest in terms of its impact on bean production in general in Southern Africa. The project has 4 objectives. The first objective is to develop and release bruchid-resistant bean varieties with good agronomic performance in market classes that are preferred in Southern Africa, that's Malawi, Mozambique, and Zambia. Then to use the existing modern breeding tools to make the selection process more efficient. And third objective is to determine the cooking time of the breeding lines with bruchid resistance. And the fourth objective is a capacity building one, which is to train the next generation of bean breeders in Southern Africa. So to introduce the project team, we have Professor Juan Osono, who's the lead PI for the project, and is based at North Dakota State University. And then we have co-PI Dr. Phil McLean, who's also at NDSU in the US. Then we have Carlos Urea, who's at University of Nebraska. And then we have Virginia Chisale, who's the bean breeder for the national program in Malawi, and Celestina, who's the bean breeder for the national program in Mozambique, and myself, Kelvin Kamfa, who's the bean breeder here at the University of Zambia. In terms of the countries that the project is focused on, we are focused on the Southern African region, specifically working in Malawi, Mozambique, and Zambia. But despite us focusing on these 3 countries, we are hoping that the outputs that will come out of this project, or the impact of the project, will kind of go beyond the Southern African region. Obviously, the bruchid-resistant lines that we develop will end up probably, part of those materials might end up with PABRA, which later on could be shared with other countries that fall within PABRA and beyond, actually, where PABRA operates from. So we are likely to have an impact beyond these 3 countries and the Southern African region. So a little bit of background to the topic that we are working on. The pioneering work of weevil resistance in common bean was done in G. Myers' lab at Oregon State University. And in that work also there were other people who were involved, including Jim Beaver and Paul Kusolwa, who's now a professor at Sokoine University in Tanzania. Originally, there was no resistance for weevils within the primary gene pool of common bean. So these guys who were really the trailblazers went outside the primary gene pool to go and look for that resistance and bring it into Phaseolus vulgaris. And they ended up getting that resistance from Tepary bean, which is a sister species of Phaseolus vulgaris. And they moved that resistance and even ended up developing a common bean variety, which possessed that resistance to the bruchids. And there is the publication that accompanied one of the varieties that they released. So these are the guys who are the pioneers of this research of introgressing or moving resistance from outside the primary gene pool of common bean and bringing it into the primary gene pool. And now, one of the things that we were asking ourselves is how transferable is this resistance and how stable is this resistance as you move it in different genetic backgrounds? And that question was answered, and the answer was yes, it's transferable, based on the research work that we had undertaken before we were awarded the project, And there is a paper that we published together with Dr. Beaver, Currency, and James Kelly to demonstrate that indeed you can move around this resistance, which is essentially conditioned by the APA locus on chromosome 4, and they can be deployed in different market classes. And that we demonstrated as we transfer that resistance from one of the resistant lines which were developed by Paul Kusola and Jim Myers into a land race which is called Solwezi here in Zambia. So once we had demonstrated that, we thought we were in a good position for us now to try and deploy this resistance widely in different market classes of beans. So that is kind of the foundation or the background to the project. And the project is focused on introgressing this weevil resistance, essentially deploying the APA locus in a variety of market classes, including the pink mottled, the purple mottled, the red mottled, and the sugar types. These are the market classes that are important and are in the major bean corridors of Southern Africa, corridors which are essentially the routes or the paths in which beans is traded here in Africa. So how do we go about with a screening for weevil resistance? We essentially use quite a simple method. We have those jars. We put the beans there, then introduce the weevils, and then assess them at 45 days after infestation and 65 days, and be able to determine how many number of perforations. And based on that, we are able to assess the resistance or susceptibility of a variety to the weevils. So under this project, what we did was now to cross these, we had selected a number of parents mainly from the Andean diversity panel, where we had a better sense in terms of their superiority in agronomic performance and also their acceptability in the countries that we are working on. And those are the genotypes that we crossed with the resistant line. And out of those crosses, we've developed a total of about almost close to 2,000 breeding lines, which is just 9 lines shy, shy of 2,000, which is the actual number is about 1,991, but there are also other lines that we have not accounted for. So when you put together, we have a total of almost more than 2,000 of the breeding lines that we've developed by crossing these genotypes with the resistant lines. And these are the genotypes that we are now screening. But we first had to screen these parents to kind of check their resistance to the weevils, and also included, of course, the resistant parents. And what this graph is showing you is essentially the reaction of these materials to the weevils. That's the parents. Not all the parents are included, but some of the parents. And what you see is there the resistant checks, which is where we are getting our resistance, which has the APA locus coming from Teperi, are quite resistant if you look at that, where the red line is the threshold. Whereas the breeding line, the other parents that we've used that are agronomically superior but are quite susceptible to the weevils, you could see how badly damaged they were based on this scoring system that we have. So we were in a good position to not say, well, the resistant parents are behaving as they should, and also the susceptible parents are also susceptible. And we were now hoping to move this resistance from these resistant checks now into the parents. So what progress have we made to date? We've made quite significant progress. And here I'm just depicting a small sample that we got out of a cross of the resistant and the ADP1, which is one of the genotypes that is superior agronomically but very susceptible to weevils. And this is out of the screening that was done here at the University of Zambia. So you could see that we're able to recover some genotypes that are the score of that had almost no perforations or very few perforations, but also we got quite a lot of lines that were susceptible. And the take-home message from this is that, yes, we are able to recover resistance in the backgrounds, in the genetic backgrounds of materials that are preferred here. However, we've had challenge that in terms of the low frequency of these resistant progenies that we get out of here. And this has been a concern because even when you have hundreds of these lines, you only recover a couple of these materials that tend to be resistant. And we previously reported about this low frequency. It's not something that we're discovering for the first time, but it's something that we've also experienced in our previous research on weevil resistance. This is not just to give you a sense of the market classes of the materials that we have recovered, the ones that are resistant. Like you could see out of this cross of 3 and ATP1, we've managed to recover these materials on your right-hand side of your slide, line 13, 28, and 51, in quite variable market classes. These have superior levels of resistance to the common weevil. In some cases, even a bit more resistant than the resistant parent itself. So this is quite exciting for us. Again, what I'm trying to show you here is just to give you further insights in terms of how variable the seed types or the market classes in which we have succeeded to introgress the weevil resistance. Some of these market classes include the purple mottled, which is a popular market class for us here in Zambia. So this has been quite an achievement for us. And these materials that are resistant together with the susceptible lines, we've planted them in our trials. What we are showing you there is an image of one of the nurseries that we have, which is at Golden Valley Agricultural Research Trust, where we have all these 1,991 that's both susceptible and resistant. We are also keeping the susceptible because we might need to use them for further genetic studies in our quest to develop molecular tools that could be used to bring about efficiency in their selection for the APL locus. So these are the materials that we have this year growing at GAT. We also have these materials at another location, which is in Malashi Mpika, and again, mainly for seed multiplication and also for evaluating them for their agronomic performance. So all in all, we've been successful, and these materials that we've identified out of our preliminary screening, we've shipped them to USA, to Dr. Wano Sono, where they are going to be planted in their, for them to be evaluated for their agronomic performance. We've also shipped them to Mozambique and Malawi, to Celestine and Virginia for seed multiplication and agronomic evaluation in those countries. The other progress that we've had is in the form of capacity building in our National Agricultural Research System. Remember, objective 4 of our project was to build capacity to develop, to train this next generation of plant breeders here in Southern Africa. And we are quite excited on this front of the project because we have 4 talented young women who the project is training. We have Maria, who's currently doing MSc studies at NDSU, and Maudrine, who's doing MSc studies in plant breeding and seed systems here at the University of Zambia. And Maria and Maudrine are from Zambia. Then we have Isabel, who's also has joined my breeding program here at the University of Zambia, doing a master's degree. She's from Mozambique. And then Rebecca, who's from Malawi, and also has joined, has started our studies here at the University of Zambia in plant breeding and seed systems. This is very exciting for us because these are talented young women, and they eventually would be in some leadership position of building research in their respective countries. Isabel and Rebecca, they are already in Zambia and they have already started their studies, and they didn't waste time. The moment they arrived, they hit the ground running. And here we're having Isabel, who's from Mozambique, helping us with our inoculation for CPP in the greenhouse. And they are currently doing their coursework. The other project activities that we are undertaking is essentially to develop marker-assisted selection for the APA locus, and this work is being anchored at NDSU. And Maria, she's the student working on that under the mentorship of Dr. Wano Sono and Phil McLean. Then we are going to undertake cooking time evaluation based on the Matson cooker and on the on-farm conditions. And we are also looking at gender integration into the project. And I just gave a presentation on that supplementary grant that we have related to this. In conclusion, we've optimized the screening protocol here in Zambia, developed a total of close to 2,000 F7 breeding lines in diverse market classes. These have been screened for weevil resistance, and we've identified lines that are resistant in very variable market classes. And seed of these bruchid-resistant lines have been shipped to our collaborators in the US, in Malawi, and also in Mozambique. And here in Zambia, we are further multiplying the seed and further screening these materials for weevil resistance and also for agronomic performance. And then the 4 students who are all female are being trained, which is very important for building capacity and really to increase the participation of women in breeding, which we struggle with for us here in Africa. So with that, I'll end my presentation here, and I have several organizations to thank: the USAID, the University of Zambia, the IIM of Mozambique, University of Nebraska, NDSU, and Michigan State, where the Legume Innovation Lab is hosted. With that, thank you very much.
Good morning and good afternoon, depending on where you are. I welcome you to this presentation entitled Screening Common Bean Breeding Lines for Bruchid Resistance. This will be a joint presentation by myself, Chinji Modrin, and Maria Mazar. We can move to the next slide. Common bean is an important crop as it is a source of protein, iron, and zinc. Despite its importance, it's faced with major constraints, which are the abiotic and biotic stresses. Among the biotic stresses, we have the common bean weevil, Acanthocleides obtectus, which is a predominant post-harvest pest in all tropical bean producing regions. On my top right, the picture shows the common bean weevil that we are working with. We can move to the next slide. Common bean weevil can cause as much as 48 to 100% losses in common bean seed quality and quantity, depending on the susceptibility of the cultivar. The weevils attack both in the field as well as in storage. Hence, it's imperative to use resistant cultivars and breed for genetic resistance is the most environmental and cost-effective means to mitigate the losses. If you look at my right down pic, it's showing the beans that has been attacked by the weevils. Hence, if a variety is susceptible, this is what the damage the weevils can do. We can move to the next slide. The objective of this study was to introduce weevil resistance within Southern African preferred market classes of common bean. The image I'm trying to show here shows the 6 market classes that are of interest in this study. We can move to the next slide. A total of 596 advanced breeding lines from 10 populations were screened at the University of Zambia and in Isanfo, that is the northern province of Zambia. The 10 populations were derived from 10 different crosses that were done at the University of Zambia. The study was conducted in a completely randomized design with 2 replications, both at UNZA and in Isanfo. AO3A and AO6B were used as our resistant checks, whereas Lusaka and Kablangeti were used as our susceptible checks. So these are the pictures I'm trying to show on my right. AO3A as our resistant parent and Kablangeti as our susceptible parent. Then the parameters that were assessed were percentage of damaged seed and perforations, the number of perforations on the 10 seed. We can move to the next slide. The slide I'm trying to show just shows the flow of activities when we are phenotyping for resistance to A obtectors. So in the first place, we need to have adult bean weevil, both the male and female. We need to have about 300 adult weevils, which are infested in 400 gram seed, so that we have a mass rearing of weevils that will be used to conduct the experiment. We expect to have images at 45 days. The seed for the experiment is first sterilized by putting it in the fridge for 48 hours so that we don't have any eggs or weevils before we do our actual screening. After sterilizing the seed, it's kept on room temperature for 24 hours. Thereafter, 10 seeds are counted and placed in the 250 plastic bottle. Then the 10 adult and sexed weevils are put in the bottle. Then they are incubated in the insectorium at 28 degrees Celsius for 30 to 60 days. Thereafter, they are evaluated at 30, 45, and 60 days. At 30 days, we remove the initial 10 adult weevils that we had infested the seed with. Thereafter, screening starts at day 45 and at 60 days, where we score for the number of perforations on the 10 seeds, percentage of damaged seed, as well as the number of image adult grouping. Hence, a seed with at least one perforation is considered to be damaged. The picture in the center, the one that has got a resistant line as well as a susceptible line. For a susceptible line, if you look at the perforation, using the CR scale of 1 to 5, if it has greater or equal to 16 perforations, it's considered to be susceptible. Whereas a resistant line, when it gets a score of 1, it means it doesn't have any perforations. They need to be given a score of 1 to be considered to be resistant. This is what the picture in the center is trying to show. We can move to the next slide. The results that will be shown are just one of the populations from the 10 populations that were screened, where we had a total of 596 lines. So in this presentation, our results are focusing on the population AO-3A by ADP 763. This is the screening at 60 days across the 2 locations. We had 120 lines that were screened. Out of the 123 lines, we had 11 lines that were resistant. They had a score of either 1 to 2. Then we had 1 line that had a score of 1. It didn't have any perforations. When it came to the parents AO3A, which is a resistant check, it had a score of 2, which shows that it maintained the resistance. Whereas for the parent ADP 763, it was susceptible and it got a score of 5.
So for the percentage of damaged seed, the population had a range of 0 to 100%, where we had 11 lines that had less or equal to 10 seeds damaged, and 1 line didn't have any of its seed damaged. And we can see that the AO3A, which was used as a parent, a resistant parent, had a score of 20%, while the susceptible parent had a score of over 90%. When this population was screened at both locations, which is Misanfo and Unza, 23 lines were resistant only at Misanfo, and 11 lines were resistant only at Unza. 3 lines were resistant at both locations. This graph is showing all the lines that were screened for the populations, and there were some lines that were resistant at Unza and were susceptible in Misanfo. Other lines were resistant in Misanfo and were susceptible at Unza, but there were some lines that were resistant at both locations. And this is quite interesting because it brings about, it could be because of different biotypes that exist in subpopulations. That's the reason why we saw a variation of these lines performing differently at both Unza and Misanfo. Out of 596 lines, 13 lines were identified to be resistant. And these lines belong to the different 10 populations that were screened, and they belong to different market classes. The 13 resistant lines that were identified suggest that the resistant APA locus in AO3A that was derived or originating from Phaseolus actifolius has stable expressions in different genetic backgrounds. These results suggest that different breeding programs can use AO3A to deploy resistance to A. obtectus to different market classes. Interestingly, there were 3 super resistant lines that had no perforations at all and had a score of 1, whereas the resistant parent AO3A had perforations and had a score of 2. The 13 lines out of 596 that were identified as are consistent with what was previously reported, that there's actually a low recovery rate of resistant lines to A. obtectus. And these results suggest that when breeding for resistance to A. obtectus, there is need to have a larger population. Right now, 1,991 breeding lines have been planted at GAD and in Pika in Zambia. And the 13 lines will be shipped to Malawi, Mozambique, and North Dakota State University, where these lines will be planted in the field to test agronomic performance and to validate a molecular marker that was previously developed. And the screening is still ongoing in Zambia. We'd like to acknowledge the USAID, Michigan State University, for providing funding for this research. Would like to further acknowledge NDSU, the University of Zambia, and ZARI, where the research is being conducted. With this, we end our presentation. Thank you.
I now welcome Dr. Kampwa to the session to answer questions. Our student presenters, Maria and Maudrine, will not answer questions live, will provide written responses to any questions submitted under 6A. But a quick reminder that the Q&A designation for this project is 6, and for the student presentations, please use 6A. I'd also like to thank all 3 presenters for those excellent talks. And over to you, Kevin, and I will. So first, I would like to welcome you to answering questions, but I'll start by giving you a question, so that will help, of course. Is there a relationship between resistance and cooking time?
We are not so sure about that, and that's one of the things that we are going to investigate. We don't know whether that relationship exists. For that, we identified those lines that are resistant, and the next thing we are going to do is to cook those lines. And then after that, we'll be able to ascertain as to whether there is any possible relationship between the 2 traits. So as of now, we haven't been able to do so.
Great, thank you. Another question was, did the crosses significantly alter the phenotype of the selected genotypes? So I assume, were there any pleiotropic effects, is essentially what they're asking? Were there other effects beyond just these?
Is the question to asking as well there are pleiotropic effects when you are selecting for vivo resistance? Is that correct?
That's how I would characterize the question. Is I guess beyond this is one very specific phenotype, which is of course resistance. Are you carrying other traits along with it that alter any phenotypes? I think that's maybe a way to put it.
Yeah, what we really had a bunch of lines to screen. We have had more than 2,000 of these breeding lines to kind of scan through and be looking at those ones that we've been successful to progress the APA
locus and see whether that resistance is holding up. Now, the next stage in which we are moving is to start screening them for a bunch of traits. This will be in terms of agronomic traits and end-use traits, including those cooking time, and it could also be nutrition aspects of that. As of right now, mainly the focus has been to say, can we recover the resistance and
I think we've lost the feed. If any other members of the team would like to, we'll just give Kevin a minute or so to come back on and hopefully continue to answer the question.
Sorry, I apologize. I think I broke off. Like you notice, I've traveled. I've traveled to a remote part of Zambia where I'm evaluating my breeding trials. So internet has been a challenge. I've had to pull over and just be able to join the session. Yeah. So there's a bit of instability. But what I was saying is, obviously now these resistant lines, the weevil resistant lines, the next step is for us to evaluate them for agronomic performance, end-use traits, which include the cooking time. And yeah, I think that's pretty much what we are looking at. Yeah.
Okay, great. Thank you very much. So I think we have essentially time for one or two more questions. Do you have any idea on which chemicals are underlying the resistance mechanisms?
It's just a cluster of genes there that are involved in coding for these toxic to the weevil itself. And my sense is that it's a combination of traits. A combination of these proteins, which are probably at play. But APA locus, it has a bunch around that chromosome 4, where the APA locus is. There is a bunch of genes there coding for these a bunch of amino acids, which are toxic to the weevils. Yeah.
Sorry.
Barry, I believe you are muted.
Thanks, Jen. What is the level of protection conferred by the resistance source at the time of storage?
I couldn't hear you. Either muted. I only got the end.
Okay, I'll turn my video off to hopefully reduce bandwidth.
And part of your question, could you just repeat that?
Absolutely. What is the level of protection conferred by the resistance source over time of storage? Oh
yeah, and what we've been able to see is that for these materials, which are the 13 lines, resistant lines, which we are very excited about. By the way, this is quite a major achievement, given that now we've been able to move them in these major market classes, including the red mottled that are popular in East Africa and also in beginning to become very popular in Southern Africa. So that has been a remarkable success. The challenge that we get at times has been the low recovery of these resistant lines. So for us to get these 13, that's pretty good as far as the project is concerned, and more important that it's in the market classes that are preferred in Zambia. We've seen that resistance holds up pretty well up to 45, 65 days, and that's very important because most of our small scale farmers are keeping the 5 days before they sell them or before they consume them. And what we are seeing in terms of these 13 lines is that the materials continue to have that resistance, or the resistance holding up all the way up to 65 days, 70 days, and in some cases even up to 80 days. And that's pretty good levels of resistance if you are.
Great, thank you. And that concludes our Q&A session. So yes, thank you, Kelvin, much appreciated. That concludes our Q&A session. Thank you, Dr. Kamfa, much appreciated. And thanks for the answering those great set of questions. So our next presentation is strategic collaborations, implementing a gender responsive genetic improvement program for bruchid-resistant dry beans in Southern Africa. This project is ongoing and led by Dr. Kelvin Kamfa, which we just heard from, and we're ready to hear from our presenting. I've already heard from our presenter on Dr. Juan Osorno's project, and Kevin will again be presenting. And the Q&A designation for this project is 7. So just go ahead and begin the session. Thank you very much, Jan. And just go ahead. Thanks.
Good afternoon, ladies and gentlemen. My name is Kelvin Kamfa, and I'm the principal investigator for the supplementary grant entitled Strategic Collaborations, Implementing a Gender Responsive Genetic Improvement Program for bruchid-resistant dry beans in Southern Africa. There are several collaborators on this project, and you'll be able to learn about them later in the presentation. So the way I've structured the presentation is that I'll give you the research background, essentially the foundations to the research, then give you a sense of the progress that we've made to date, the lessons that we've learned, the steps that we are making. Research this new supplementary grant was awarded to us in 2021. And the background to this was that it really was building on in terms of the increased capacity that some of our team members had obtained out of the training workshop that was organized by the Legume Innovation Lab. And it was offered to the team members by the GREAT team, which is essentially a gender responsive researchers for agricultural transformation, which is as the acronym of GREAT. So during that workshop, training workshop, which was funded by the Legume Innovation Lab, there was capacity that was built in terms of gender responsive plant breeding. And there were work plans that were also developed, and some of the topics that were covered in the sessions are presented on that slide. But in a nutshell, quite a lot of capacity was built from that in terms of gender responsive plant breeding. And we wanted now to build on that and ride on that capacity that was built to try and now mainstream gender a responsive breeding in our programs here in Africa. Also the background to that, we thought a project on gender responsive plant breeding would be an opportunity to respond robustly to the guidance that was given to the Innovation Lab by USAID in 2018 to try and mainstream responsive gender responsive legume research in our projects. The project brings together a talented team of gender advisors from the host countries, the ones that are involved in the project, which include Zambia, Malawi, and Mozambique. So the project was guided by a number of research questions, and we really wanted to develop a better understanding in terms of what are the factors that are involved in terms of mainstreaming our breeding work. But the guiding principle for this was that gender analysis in legume research needs to articulate the roles of women and men in legume systems and how these roles inform research and processes and pathways to impact. Then identify the relevant gaps in the status and anticipated participation of women and men that could hinder our activity outcomes. Then anticipate the possible differential effects the activity may have on women, men, and youth, and then generate recommendations for meaningfully engaging women, men, and youth in legume research, and then suggest the ways that the activity could narrow, close the gender gaps, address the inequalities, and empower women. Like I earlier mentioned, this project is being implemented in 3 countries, including Zambia, Malawi, and Mozambique. And I'm just a PI, but I'm working with talented gender experts from these 3 countries. And what I'll do now is to give you a sense of the people who are involved in this project. So from Mozambique, we have Maria, who's our gender advisor for that. And she's leading our activities in Mozambique. And she's from the Mozambican government there. And then we have Hilda, who's leading our activities in Malawi. And she's our contact person for all the gender-related activities in Malawi. Then in Zambia, we have Krisanti Ichama, who also works as a gender advisor for the Zambian Bean Network. We've been privileged to have Krista Isaacs, who's an assistant professor at Michigan State University, as our gender expert. And she brings a wealth of experience in terms of the application of the gender responsive breeding tools to our project. So we are quite happy to have her on the project. The other person on our project is Andrea Allen, and Andrea works as the gender advisor for the Innovation Lab. And she has been very instrumental to the implementation of the project. And I must really thank her, and she has done a tremendous job in terms of doing all the literature search that we needed. She's the one who coordinated that, and also trying to develop the research design and also develop the tools that we need. And she's the one who has been very instrumental in kind of bringing all of us together and be able to undertake these activities. She has also been serving more or less like a project manager in the early stages of the project, and we are so thankful to her for the help that she has been providing to the project. So what is what progress have we made today up to date? And so we have in the project almost 8 research questions that we were trying to address. If you look at those 8, the first 4 research questions and part of number 5, they essentially try and help us explore the gender dynamics that are involved with regard to the producer's needs and also the preferences. That's what those first 5 research questions are trying to address. Then question number 6 up to 8, they try to explore the issues that are involved in the operations of associated breeding programs. So these are the 8 research questions that we are trying to address in our project. Now, to address these research questions, we need tools to do that. And we've been fortunate that recently there was a publication of the gender responsive breeding toolkit, which essentially tried to help in terms of developing what we might call customer and also product profiles. So that's the tool that we are using here in the project. Now, what we did at first was to try and use the existing secondary data on bean customers in the 3 host countries in Zambia, Malawi, and Mozambique, and then try to organize that customer data within the analytical framework of the gender responsive breeding. And then thereafter, we did identify significant gaps that were in that data, and those gaps were related to the socioeconomic, gender, were related to the demographics, and also socioeconomics, and also the agroecological aspects of those customers. So we had to fill in that gap. There was need for us to fill in gaps so that we could now end up developing the customer and product profile. So to do that, we've had to develop now tools to collect that data, and I'll be able to share with you a bit more in terms of the type of tools that the project has so far managed to develop for us to be able to collect that data. These tools essentially are about, include the surveys and also the focus groups, which will be able to provide us with data that could help us have that set of information that we need for us to develop the customer profile for different market segments. And also we will have the participatory variety selection tools, which will help us have a complete set of information that we need for us to develop the product profile. So these tools have already been developed. They are being fine-tuned by the project members. So this has been quite a progress, because at least now we have tools to collect that data which was missing in our secondary data, for us to be able to develop a better picture of the product and the customer profiles. This is just a work plan which tries to give a sense of the tasks that we have accomplished to date, and in terms of where we hope to be moving forward up to next year. Like you could see there, we've already done the literature search to have a better understanding, and also we have constructed which could help us develop that customer profile. And these activities, these tasks in green, some of them have already been accomplished in 2021, and others we are working on them in 2022. What we are hoping now, once we've now developed these tools to collect additional data, it be through surveys, focus groups, and also the participatory varieties selection tools, we hope then to pilot them here in Zambia and further fine-tune them so that they could eventually be adapted and applied in Malawi and Mozambique for us to collect the data that we need to develop the customer and product profile in these countries. So this is just now an outline of the work plan, where we hope to be in the next 1 year and where we are coming from. What lessons have we learned to date? One of the challenges that we had to deal with was COVID-19. We had made very good progress at the beginning, but due to COVID, there were restrictions in terms of the movements within countries. And especially in the case of Zambia, our gender advisor couldn't go out in the field to try and pilot the use of these tools that we've developed. There are also challenges in terms of Cyclone Ana in Mozambique and Malawi. The other challenges come in terms of for us who are administering this project, specifically for us at the University of Zambia. There has been a learning curve in terms of from the time we had to create the project, in terms of up to the time the paperwork had to be processed, and eventually the project awarded, and then I started receiving funds. That took a little bit of time, and there were a few, we lost a little bit of time. So that was also a challenge by itself. What are the next steps that we are taking moving forward? So we've made remarkable progress in terms of first understanding the gaps in terms of information that we needed to develop the product and customer profiles for common beans in Zambia, Malawi, and Mozambique. And we've developed the tools that could provide that information that we could later use together with existing information for us now to develop those profiles. So what we now need to do moving forward is for us to be able to pilot these the data collection tools, and this will be done through the field days and also participatory varietal selection in our various nurseries here in Zambia. Then we need to complete or revise the gender breeding customer profile, and also consider the implications for the gender breeding product profile, and then adapt these tools for use in Mozambique, Malawi, and to continue using them here in Zambia. So these are the next steps that we are hoping to undertake. So at the end of this, the output that we are looking for is that at the end of this project, we could have our research, the capacity for us breeders in terms of using gender responsive breeding could have been enhanced. Essentially through our participation in the application of these gender responsive breeding tools. We hope to produce one working paper and one manuscript for publication, in which we'll be able to share widely with the research breeding community in terms of the application of gender breeding activities in our breeding programs. For the people who are very familiar with this work, and Krista Isaacs is one of them, who's the assistant professor at Michigan State. I think we are happy with the progress that we've achieved. And just quoting her there, she says the team has done a remarkable job in figuring out the gender responsive, breeding tools and pushing forward with these activities despite the challenges of COVID, and we are looking forward to the research output. So we've made, I think, steady progress even in the middle of challenges such as COVID-19. And we are looking forward to provide you with results at the next convening. With that, I would like to end my presentation here. You can contact me for further information. My details are provided there.
We'd like to thank Dr. Kampfa for that excellent presentation. The Q&A designation for this project is 7. I'd like to reiterate that. And I now welcome back Dr. Kampfa to the session to answer questions. And we don't have a significant amount of time for questions, so we'll basically allow for one question. Did you find significant differences between men and women in terms of preferences for bean varieties?
I think and this is when we are going and also collecting data using these focus group discussions, and that's when we'll be able to determine as to whether there are these differences, which some people have said that anecdotally there is some evidence that indeed there are those differences, but we are excited that I think differences exist through a more structure, mainly based on the secondary data, the existing data that is coming from Zambia, Malawi, and Mozambique, and that data has those gaps. It doesn't really have the numbers disaggregated between gender and these tools will be able now to determine exactly whether those differences do exist or not. And other gender experts, they are in the audience, and please feel free to chime in to try and provide more context to my answers. Yeah, but that's where we are now.
Okay, great. Thank you. If anybody else from your team wants to weigh in, we have about a minute before we need to move to the next section. So
here's one. I can probably add to it a little bit. The short answer is yes, because hey, I'm here. Yeah, okay. The short answer is yes, there might be differences. We have seen cases in the past of varieties that had a lot of the good agronomic qualities and characteristics that we were looking as breeders. You know, the production part was really well done, but then when it comes down to the cooking part, the kitchen, the sensory, we found that there were issues, and the variety pretty much died because of it. So that's hopefully one of the things that we really want to make sure that we're answering. That's one of the main questions we want to answer here is, okay, in many cases, we know that the breeders are aware of these things. They're aware that, you know, there are certain preferences between males versus females, and the cooking time, of course, is a big deal for females. But even though we are aware of that, why at the end for the decision-making process, those things kind of disappear from the radar, right? And what needs to be done in order to make sure that we still are accounting for cooking time, for sensory, for taste, for all these things. You know, the color of the brine sometimes is a big deal in some countries. So yeah, that's one of the things that I'm as a breeder, I'm really interested in knowing what are the factors that are affecting that decision-making process that at the end of the day, sometimes we forget about them, among many other things, many other traits that we're trying to figure out, right?
great, thank you. And I'd like to thank both Dr. Kamfwa and Dr. Juan Osorno for answering those questions. Much appreciated. So thanks, Juan, much appreciated. Thanks, Kevin. So our next presentation is transforming seed systems to respond to bean variety demand through multi-stakeholder platforms in Malawi. This project is ongoing and is led by Dr. Jean-Claude Ruboyo from the Alliance of Biodiversity International and CIET. Presenting today is project team member Dr. Eliud Barichi, and the Q&A designation for this project is number 8.
Good morning, good afternoon. My name is Eliud Barachi from the Alliance of Biodiversity International and CIET. I'm going to share with you the project updates on behalf of our team. The project is referred to as Multi-Stakeholder Platforms Project, which is in the full name, it is transforming seed systems to respond to bean variety demand through multi-stakeholder platforms in Malawi. To guide us through this presentation, here is an outline. We're going to look at the introduction, and then look at the research to date and some of the research findings. Look at inclusion of cross-cutting themes and scaling and technology and policy maker engagement plan. The project works in Malawi in 3 districts. 2 of the 3 districts are what we refer to as intervention sites, and 1 is the counterfactual or control sites. The intervention sites are the districts where the multi-stakeholder platforms were established, while in the counterfactual sites, there were no multi-stakeholder platforms. So the districts are based, or they're located in the northern part of the country and the central region of the country. As part of the team, we have a number of partners, and listing a few major ones. We have Tradeline Corporation, Muzusu Catholic Development Commission or CADECOM, Virginia Tech, Department of Agricultural Research and Services, the Lilongwe University of Agriculture and Natural Resources, the Alliance of Biodiversity International and SEAT, seed companies, grain traders, and produce organizations, among other partners that we work with. The project works around 3 main objectives, and each of these objectives targets certain deliverables. In the first objective, which is about generating evidence on the role of MSPs in increasing investments in certified seed supply, this objective focuses on seed supplies and specifically the certified seed supplies. The project has been able to engage different partners in the seed supply system in the country through some meetings that have taken place virtually and face-to-face. In this way, we've been able to generate partner profiles. We have also been able to map out these stakeholders in a gendered manner. For example, in the last session that we had with partners, we had 10 women and 15 men comprising of that meeting. We have also been able to narrow down on the varieties and the profiles. These have been agreed upon by the stakeholders. In addition, different classes of seeds are being multiplied, that is pre-basic, basic, and even certified seed. And these are expected to end up with the private sector partners. We have been able to recruit 3 students in the project, 2 males and 1 female. Each of these students is focusing on one objective. So in objective 1, we have 1 student that is research or carrying out research on the role of the multi-stakeholder platforms on the supply of the certified seed. The students have developed their research proposals, and these are awaiting the presentations to the faculty, which will authorize them to proceed to the field for data collection. Data collection tools have been developed in readiness for the surveys. This data will be used for the baseline conditions as well as meeting the respective student research needs. In the meantime, secondary data and reviews on seed challenges are being conducted. Now, in the second objective, which is focusing on generating evidence on whether an MSP approach does or does not increase certified seed purchase by men, women, and youth farmers, and the extent of effect, if any. So this objective focuses on driving the demand for certified seed and is looking at the role of the MSP approach in this in generating or affecting the demand for these seeds. So in this objective, as I mentioned earlier, we have also one student who is really who is studying the determinants of demand for certified seed and has already prepared his proposal. The activity has been able to generate lists of key stakeholders, and their roles have been prepared. So we have about 9 seed enterprises, 3 grain off-takers, and research and extension stakeholders. These lists are now available based on their active participation. Varieties that will be used for promoting for the promotion using the small packs have been identified. 4 major bean types of interest in this activity. They fall under the classes of red mottled sugar, small white, and red kidney bean types. 4 seed companies have also been recruited, or essentially they have agreed to participate in small packs marketing as a way of promoting these varieties. In addition, 4 demonstration plots have been set up, and 56 farmers, but 1 of them female, are learning from these demo plots that have been set up. In our third objective, which is looking at evidence that using the MSP-based PV, or participatory variety testing approach, increases awareness and accelerates uptake of new market demanded varieties by men, women, and youth. So this objective focuses on the new varieties and some of the most promising but a little older varieties. So, and it's looking at what is the role of the multi-stakeholder platform in being able to raise awareness and accelerate uptake of the new varieties. So we're working with the existing varieties, but we anticipate some varieties to be released in the course of the project, and therefore these approaches will be applied to them. So to be able to achieve this objective, 4 testing sites were identified, and these are in different locations. And for these sites, they were the 4 bean types that I mentioned earlier in objective 2 have been agreed upon. So the specific varieties that fall under these 4 major bean types or classes have also been identified. And in total, we have 15 varieties that have undergone the trials at the 4 sites. Of these 15 varieties, 5 are newly released, but 10 are older but promising varieties. We also have 39 demonstration farms that have been established, and this will be used for variety evaluation purposes. There are also 4 on-station trials that are located in 4 locations that have been set up, and these ones are linked to the national research under DAS, or Department of Agricultural Research and Services. So we have a student in this objective, and she has developed a proposal and will be studying in depth on the effects of this MSP approach on the rate of awareness and uptake of these market demanded varieties. So far, the project has not achieved a lot of updates or outputs or full outputs per se, but in the last few months that the project has started operating, we are seeing some partial outputs or emerging outputs, and this we can name a few of them. The initiation of the projects and the beginning of its implementation was a major milestone. In addition, the recruitment of the MSc students who have gone on to develop their research proposals is also a milestone for the project. The engagement of the partners in the project is also another milestone that has been achieved. We have lists of partners and their profiles that have also been developed. We also have lists of market demanded varieties and their profiles. In addition, the tools for both baseline and student data collection has been developed and is ready for use. And finally, project demonstration farms and trial sites were selected by project partners, and these have also been established. Now, in the next 6 months, we have a number of priority activities which are in line with the project work plans, and these include the following: completion of the surveys, students to analyze data for their thesis from these surveys, evaluation frameworks and indicators will be refined. The multi-stakeholder platform learning meetings will be conducted. The digital landscape and interventions will be put in place. Seed production for winter crop and supply to private sector will also be established. The use of small packs as a marketing tool will also be implemented. Demonstrations and awareness sessions and training of facilitators for crop management or bean crop management will also be conducted. Monitoring and evaluation data will continue also to be collected. And finally, we anticipate to generate some emerging success stories from implementation. So among the cross-cutting issues, which focus mainly on inclusion, we have first and foremost gender inclusion, which is being promoted at different levels, whether it's there is the overall mainstreaming within the project, in the recruitment of students, in the platform leadership and memberships. We are focusing to have gender inclusion as a key theme or issue. Digital inclusion among producers is also a key focal area by the project, as well as financial inclusion for women and poorer producers. And we are also deepening value chain participation by men, women, and youth within the bean value chain. Now, with respect to scaling and technology engagement, scaling the technologies, as well as policy engagement, we have some plans, and these plans revolve around the MSP, the multi-stakeholder platform that has the different stakeholders already actively participating. Among these include producer organizations, traders and processors, input and services providers, policy and extension service providers, development agencies, among others. So these are engaged in the project in order to support the uptake and scaling of these technologies and innovations. And we have on the ground partners such as Mzuzu Cadecom and Tradeline Corporation that are actively involved in scaling efforts, working with the entire project team. For example, Mzuzu Cadecom works with the producers and facilitates the MSPs, which also interface with the government institutions in the different regions. Tradeline, being a private company, facilitates market access, digital inclusion and trade, and microfinance operations for the platform and the stakeholders involved. So these, together with the government engagements, are designed to ensure that we are able to scale these technologies further. So the photo here is an indication of one of the meetings, the multi-stakeholder platform meetings, one of the first face-to-face engagements in the country. This comprises all the different stakeholders I've mentioned, and they are fully engaged in the implementation of the project. We very much acknowledge the support from the USAID with the future program through the Innovation Lab for Legume System Research under the Michigan State University. Thank you very much for this support.
Great, thank you for that excellent presentation. I now welcome Dr. Ryobiogo and Eliud Barachi on to the session to answer questions, and we'll probably only have enough time for one or two questions. So let me just begin with the first question here, and it's for the control or counterfactual areas, how do you determine whether there are already existing multi-stakeholder platforms or not in the areas you're working in? Thank you.
Yeah, thank you for the question. So the definition of the multi-stakeholder, whether it's one or not, this depended on those that, the implementers are already on the ground. The project team is already or was already aware of what is happening with respect to multi-stakeholder platforms. So, and they were aware that in one of the districts, we did not have these platforms. At least we were part of those establishing these platforms, and they have been running as part of the ongoing programs. So there was prior awareness of where these multi-stakeholder platforms are located in the country based on the experiences and knowledge within the project team.
Oh, great, thank you very much. So the other question is, how is the project's linkage and or alignment with other seed system initiatives and other MSPs in sub-Saharan Africa? Do you have linkages with other programs that are doing similar types of things? And if so, could you elaborate?
Yeah, thank you. Yeah, there are other multi-stakeholder platforms that the teams have participated in before. For example, we had the Sub-Saharan Africa Challenge Program under FARA, that is Forum for Agricultural Research in Africa. So these were among the initial multi-stakeholder platforms being established across Sub-Saharan Africa. And we also have some even in Southern Africa, even within Malawi. So these multi-stakeholder platforms that we are working with in Malawi are actually among some of those that were previously established. So this project did not establish any platform and is not going to establish any, but it is using the existing platforms that have been established some years ago and really trying to use them to catalyze the work on seeds. And they've been also efforts, on seed work using platforms. And I think let me call on Jean-Claude to also give some aspects of that.
And I just said we have a hard stop in about 1 minute. So, but go ahead. Thank you.
Yeah, maybe thanks, Eliud. Thanks, Barry. Yes, the Pan-African Bean Research Alliance has been able to work with the grain market traders and national program, and you have the bean corridors, and these are also within the corridor they embedded the platforms based on off-taking and the grain market and traders. So this again will allow to share experience across the regions, across the countries, and across the different market types, and compare the success and learning lessons, take lesson as well. So we're looking toward other crops too. So that's not only beans, but also other crops as well, through other initiatives too, yeah.
Great, wonderful. And I'd like to thank both of you for that excellent presentation and for answering those questions. Highly helpful. Very interesting work, much appreciated. So we're now going to take a short 9-minute break. We'll see you back in 9 minutes, and the research session will begin promptly at 11:40 a.m. Eastern Standard Time. So we'll see you in 9 minutes. Thank you very much. And I'd like to thank all the presenters this morning for those excellent talks. Much appreciated. And thank you very much, and we'll see you in a little while.
Welcome back from the break, everyone, and greetings. My name is John Maidendorp. For those of you who are joining us for the first time today, I am the Deputy Director of the Legume Systems Innovation Lab, and I will be moderating the remainder of the sessions today. Our next presentation is improving incomes and nutrition security through development and commercialization of consumer-preferred processed legume-based products in Malawi and Zambia. This project is ongoing. In fact, it's just getting started, and is led by Dr. Robert Fungo of the Alliance of Bioversity International and SIAT. Presenting today is Dr. Fungo, and the question and answer designation for this project is the number 9. That is the number 9.
Good afternoon, good morning to all colleagues across the globe that are attending this global meeting. My names are Robert Fungo. I work with the Alliance of Bioversity International and SIAT. In particular, I work with the Pan-African Bean Research Alliance, which is hosted by SIAT. I am a nutritionist by profession. I sit in Kawanda Research Station in Kampala city of Uganda. I'm here to talk about the project that has been awarded to PABRA and SIAT. We have been awarded a project to basically to improve on their incomes and nutrition security by developing and also supporting commercialization of consumer preferred processed legume-based products in both Malawi and Zambia. And this work will be done by a consortium of partners across the globe. We have got partners in the US, Virginia Tech University, that will be supporting some of the socioeconomics work in this project. The Michigan State University colleagues who are the ones backstopping us. And then we have partners in Zambia, the Zambia Agricultural Research Institute, partners in, Zambia again, the University of Zambia, the Agricultural Research Institute in Malawi, and the University of Lilongwe in Malawi. We will be working with all this big team to deliver this project. This this presentation is, this outline has been provided by colleagues at Michigan State University, and I'll follow that recommendation they have given us to present our work according to this outline. Briefly, I'll talk about who we are working with and where we are working. I'll talk about the recent the update where we are with the research. I also talk about cross-cutting issues and the issues to do with sustainability and scaling that we think can scale this project after it has ended. Because it has recently been approved, this project, it was approved around December, but the signing of agreement came in late. It was actually about a week ago that we signed the agreement. So you can imagine in the context that we work in the region, some partners were hesitant to proceed without the contract being signed. So we said there's some bit of delay, but we have something to share with you. The overall goal and the project, the goal of the project is basically to catalyze value addition. Basically, is to, catalyze value addition for nutritious legume foods, and also analyze the preferences for existing and the potential future legume-based processed products. Along the way, we shall identify the product concepts that are relevant to different consumers. Once this is done, we shall inform two specific objectives. The first objective is to increase access to evidence-based information on consumer preferences and demand for processed legume-based products. The second specific objective of this project that we are working on is to increase the access to consumer acceptable, nutritionally enhanced, and safe optimized legume-based products. I will not talk about the partners. I've already talked about them in detail. But in terms of location, the project is located in Zambia and Malawi. Malawi is this strip, tiny strip of land. And Zambia and Malawi are located in the southern part of Africa, for those who don't know. And basically, they are agricultural countries, and they consume a lot of legumes, and they are processed products that are on small scale in both countries. That's why the project is focused in those two countries. And up to date, the current research that has been we have done, basically it has been mostly to do with preparatory meetings and developing tools that have been implemented for the two objectives. The first objective, under the first objective, we have held virtual meetings with our partners in both countries, including the USA partner, to share project expectations and also to get feedback from partners, what they expect from us. The second activity that has been done, we have developed tools and tools for the rapid markets assessment study. The tools are ready, and the field work will be starting very soon. We have gone further with partners in both countries to map the processors and the products they are producing. This will assist us in implementing the rapid markets assessment and even other studies, because once we know the processors and the products they are producing, we shall not delay in implementing the studies that we want to implement in those two countries. To support these activities, we have identified 4 master students. They have gone further to develop their research concepts, they are developing the research concepts, and they will be approved by the project very soon. Regarding objective 2 activities, again the same. Virtual meetings were held with our partners to plan the implementation of objective 2 activities. Tools and protocols for identifying the products in the field, collecting the samples, storing the samples, and then transporting the samples to the lab, nutrition lab at Lilongwe University in Malawi, have been developed. They are ready to be deployed. Field work to be carried out very soon, and then field work and lab work to be carried out very soon once we commence implementation. And again, to support these activities, we have mapped the SMEs and the products they are producing in the 2 countries, and 2 more master students will be supporting this objective. They have been identified and recruited. The students have gone further to develop their concepts and protocols, and we shall be approving them very soon as a team. The planned activities in the next few months before the project ends include We are planning to have an inception workshop to launch the project, most likely the first week of May, or if all goes well, we could have it the last week of April. Secondly, we shall conduct a study on analyzing the nutritional quality, functional properties, shelf life, and safety attributes of the legume-based products in the two countries. Immediately after this activity, we shall conduct conjoint-based analysis studies, and then we shall identify, collect, prepare samples for optimization. Immediately the conjoint is done, immediately once we have profiled the products from the two countries, we shall go further to screen them, and during the screening, we shall know which products are most preferred. And these are the ones that will go for the stage of optimization. And there are quite a number of optimization studies, activities, sub-activities that will be done, including optimizing the formulations and recipes, and then developing the prototypes in preparation for experimental auction study. Before September, we anticipate also to do a policy review, policy and regulations and standards review for both countries for processed legume products. And at PABRA, we shall develop terms of reference and recruit the consultant. And then the consultant will go further to do the reviews and the travel around, travel in the two countries to meet the processors and the policymakers to establish gaps and propose recommendations. These two thought will be done before September, but there are more activities after this. Gender equity and gender equality, equity and inclusion, yes. We believe that product will adopt and the rich benefit and empower a section of women that participate in this value chains of legumes. And we shall monitor and evaluate gender outcomes at the end of the project. The youth, yes. Africa has the youth population is very high, more than in some countries, more than 50% of the population is composed of youth. In those two countries, the youth population is very high, and we don't want to take chances. We want to support some SMEs that are owned by the youth, and we believe if we do that, we shall support some entrepreneurship skills, especially when we work with the National Bureau of Standards to standardize the products of these SMEs that we're going to, or processors that we shall identify in both countries. At the end, we so believe that we shall create employment for both the youth in and outside agriculture pathways. In terms of scaling, the project will apply an integrated transdisciplinary research design. This approach allows continuous engagement. We shall be engaging our stakeholders right from the beginning, right from the beginning up to the end. And this will be done in consultation, because optimization of products requires input from stakeholders throughout, and any stage that requires any particular stakeholder, he or she will be consulted to make input in the product development. And once that is done, the research in the project is to ensure that it's appropriately designed and implemented. And the research products are developed jointly with stakeholders. Yes, I've just talked about that. Then in terms of scaling, we are going to produce protocols for industry to produce shelf-stable legume products. And also, as I talked earlier about policy review, we shall review the current country policies, regulations, guidelines, and they have some document in place identifying gaps and recommendations. This, we believe, can support the scaling. Someone can come and pick up the project to scale it up in the two countries. Thank you so much. That is the project I've presented. We acknowledge the support from the USAID and support from the Feed the Future Legume Lab for Legume Systems Research, which is hosted at Michigan State University. Thank you.
Thank you, Dr. Fungo, for that presentation. We have a couple of questions for you. If you would please join me on camera and microphone, then we'll address these questions. Yes, thank you. There you are. This first question is one that looks like it's already been partially answered, but maybe you can elaborate a little bit on the process. How will the identified promising products be produced? Are you working with the private sector to identify a possible manufacturer?
Yes, we will be working with the private sector right from the beginning to identify the products and also work with them to optimize the products in case they have been identified that they are preferred.
Mm-hmm.
So we will be working with the private sector in both countries, then go to the lab, do the nutrient analysis, then work with the private sector to optimize the product using their facilities. And here we shall get support from the National Bureau of Standards of Malawi and Zambia to make input as we work through the optimization process.
Understood, yeah. So a second question is, you mentioned working with youth entrepreneurs. How will the project identify the youth, and will youth without processing equipment be able to work with the project?
We have, as PABRA, we have processors in Malawi and Zambia. Some of them are youth-owned, they are youth who are owning them. Some are female-owned. And if this project will give a chance to some of these youth that fit into the project objectives to select their products. Then by doing that, we shall be supporting the youth. And this has a multiplier effect, because if we support the youth, we know that we are building his skills, and then in long term, they will be able to support their families, also scale their business, earn some money. Yeah, we will be working with the youth, and we have a list. We have already identified some of the youth-owned SMEs in both countries, and it's not starting. We're not starting from scratch. We are beginning. I mean, we have a list already. We have been working with them already, and this will be a plus for them because they have got support from us before. So we believe this project will scale some of the activities we have been implementing in the two countries, and it will make the youth and the women in both countries that are processors more competent, have safer products, and hopefully they'll scale on their income.
Mm-hmm. And then one final question, Robert. Will these be new products only, or will you also be looking at enhancements to existing products?
Yes, we will be enhancing. We will be enhancing on the quality of the existing products. That's what I mean. We already have these products in the two countries, but now we are going to enhance on the quality of the existing products. If there's any new product and it is really interesting, we shall also work with them.
Okay, and enhancing them in what way? Enhancing their nutritional value, enhancing their flavor? What is the intention? Did you hear me, Robert?
No, I didn't hear you.
Oh, sorry, I'll repeat. And enhancing them in what way? Will you be enhancing the flavor, enhancing the nutrition?
Okay.
What are the enhancements?
Right, yeah. Yeah, it is the enhancement. There are quite a number of parameters that we shall work on. One is the nutritional quality. Two, the safety and the shelf life will be worked on. The functional properties, the texture, the mouthfeel, some of those functional properties will be worked on. Then the microbial quality, it is related to safety as well, will also be undertaken. I mean, we'll conduct studies to improve on that. But still, my colleague Inid Katunji will be guiding us on the socioeconomic side. I request her to also join. I mean, she can also respond to some of these questions. She's part of this meeting.
Yes, and I'm afraid we don't have time for that right now, but perhaps she could type in some answers into the question and answer section of the panel. So thank you, Dr. Fungo, for your presentation and also for answering those questions. Before we begin our next presentation, I'd like to take a minute to mention the cross-cutting themes once again. To assist each project in addressing the 5 cross-cutting themes of gender, youth, nutrition, human and institutional capacity development, and resilience, the Legume Systems Innovation Lab has created the Resilience Resource Team, or RRT. This group of expert advisors provides cross-cutting activity support to lab projects through a series of competitive supplemental grants managed by the management entity. Yesterday, we saw a short video, or a couple of short videos actually, on gender and nutrition. Continuing today, we will share short video highlights of the work of our projects through these supplemental grant awards awarded to date. We will take a few moments now to view a short video on the Legume Lab's human and institutional capacity development efforts. Our next research presentation is quantifying the scale and scope of nutritious cowpea products in local markets in West Africa. This project is ongoing and is led by Dr. Véronique Thériault of Michigan State University. Also, please note that Dr. Thériault's presentation is recorded in French, so a reminder to our English-speaking participants to ensure you have selected English in the Zoom language channel. Presenting today is Dr. Thériault. The question and answer designation for this project is 10, the number 10.
Bonjour ou bon après-midi à vous tous. C'est avec grand plaisir que je présenterai au nom de toute l'équipe les réalisations faites et à venir sur la quantification de produits à base de niébé sur les marchés locaux en Afrique de l'Ouest. Tout d'abord, laissez-moi vous présenter notre équipe. Je suis Véronique Thériault de Michigan State University. L'équipe est composée de Dr. Melinda Smale de Michigan State University ainsi que de Dr. Aliou Diagne de l'Université de Thiès au Sénégal et de Dr. Mamadou Sissoko de l'Université des sciences sociales et de gestion de Bamako au Mali. Le but de ce projet est de promouvoir un secteur du niébé plus diversifié et qui offre plus d'opportunités d'emploi, de revenus et de consommation, et ce, tout en créant un environnement propice pour les divers acteurs de niébé, y compris les petites entreprises dirigées par les femmes sénégalaises et maliennes. Spécifiquement, les objectifs du projet sont de caractériser l'échelle en termes de quantité et de localisation géographique, ainsi que la gamme de produits à base de niébé commercialisés dans les marchés centraux et secondaires du Sénégal et du Mali. Deuxièmement, de mieux cerner les changements de préférence des consommateurs et le niveau de consommation actuel des divers produits à base de niébé offerts dans ces marchés. Troisièmement, d'identifier les opportunités pour accroître la demande pour les produits de niébé à valeur ajoutée tout en mettant un accent particulier sur ceux ayant un contenu hautement nutritif. Quatrièmement, de déduire des implications pour un engagement potentiel du secteur public et privé dans la production et la commercialisation de produits à valeur ajoutée au sein des différents maillons de la chaîne de valeur de niébé. Et finalement, de renforcer les capacités des différents partenaires locaux à conduire des études de marché et de consommation rigoureuses et à mettre en œuvre des recherches sur les politiques qui reposent sur des méthodes et des outils innovants. La dernière année a été principalement consacrée aux activités concernant la caractérisation de la portée et de la gamme de produits à base de niébé offerts sur les différents marchés au Sénégal et au Mali. Des questionnaires pour caractériser les marchés, les vendeurs de niébé ainsi que les produits faits à base de niébé qui sont disponibles aux consommateurs ont été élaborés, pré-testés et ensuite réalisés dans les deux pays. Au Mali, les enquêtes ont eu lieu auprès de 400 vendeurs de produits de niébé localisés dans 26 marchés des 5 régions du sud du pays, soit à Bamako, Kaï, Ségou, Koulikoro et Sikasso. Au Sénégal, les enquêtes sont représentatives au niveau national. Environ 1000 vendeurs ont été questionnés dans 100 marchés répartis à travers les 14 régions du pays. Au Mali, les enquêtes terrain ont porté directement sur les vendeurs de niébé, tandis qu'au Sénégal, les vendeurs de niébé ainsi que ceux qui n'en vendent pas ont été interviewés. L'analyse de ces données nous permettra de mieux cerner les caractéristiques qui influencent le choix de vente de niébé ou pas. Suite aux enquêtes terrain, les équipes maliennes et sénégalaises ont produit plusieurs rapports de mission. Dans chaque pays, 2 étudiants à la maîtrise ont participé aux enquêtes terrain. Au Mali, les 2 étudiants ont déjà complété avec succès leur mémoire de maîtrise. Au Sénégal, les étudiants rédigeront leur mémoire cette année en utilisant la base de données collectée. Dr. Aliou Diagne investigateur principal au Sénégal, a travaillé très fort à développer des livres de code pour les données en français et en anglais, et ce, afin de faciliter l'accès et l'analyse des données collectées. Une série d'études de recherche utilisant les bases de données collectées sont en cours. Au Mali, Dr Mamadou Sissoko a terminé une étude qui porte sur le potentiel de développer les ventes de niébé au-delà des grains dans les marchés locaux. Cette étude a été soumise à une revue scientifique pour publication. Voici un sommaire des résultats clés de l'étude conduite par Dr Mamadou Sissoko. Parmi les produits transformés à base de niébé, on retrouve les choux froufrous, les akras, les choux boulettes, le fari, ainsi que le niébé cuit. Parmi les autres produits de niébé vendus au marché, on y retrouve aussi les feuilles fraîches pour la consommation et le fourrage pour le bétail. Les produits transformés à base de niébé sont quasiment exclusivement vendus par des femmes détaillantes, tandis que les grossistes et collecteurs de grains de niébé sont exclusivement des hommes. La contrainte majeure pour les vendeurs est l'approvisionnement en grains qui demeure difficile, surtout en basse saison. Les résultats des analyses économétriques indiquent que les vendeurs qui se spécialisent dans la vente d'un seul produit, par exemple les femmes détaillantes qui ne vendent que des beignets de niébé, ont des marges de profit plus faibles que les grossistes hommes qui vendent du niébé, mais aussi d'autres grains. La marge de profit par unité de produits transformés est plus élevée. Cependant, cela ne veut pas dire que la vente de produits transformés est plus rentable, car cela dépend des quantités qui sont vendues. Grâce à sa participation à ce projet, Abdoul Kader Traoré a non seulement acquis ses premières expériences terrain, tout comme les autres étudiants du Sénégal et du Mali, mais a aussi pu compléter son mémoire de thèse. Toutes nos félicitations, Abdoul. Le but de son mémoire était d'analyser la distribution spatiale de la production, de la transformation et de la commercialisation du niébé au Mali. Comme indiqué sur la carte en haut à droite, les régions de Ségou et de Koulikoro sont les grands bassins de production de niébé. La plupart des associations de transformation des femmes sont situées dans les régions de Ségou et de Mopti. La transformation traditionnelle avec peu d'équipement domine. La consommation de niébé est plus grande dans les régions rurales qu'urbaines et les grains de niébé rouge sont plus dispendieux que les grains de niébé blanc dus à leur rareté. Demba Diallo a aussi terminé sa maîtrise et grâce à son expérience terrain acquis au cours de l'an dernier, il a réussi à se trouver un emploi comme enquêteur avec l'INSTAT, l'Institut national de la statistique au Mali. Toutes nos félicitations, Demba. Le mémoire de Demba portait sur les déterminants de la consommation de niébé par les ménages maliens. Les résultats de ces analyses démontrent que la part des dépenses alimentaires allouées au niébé demeure faible, surtout en comparaison avec les autres denrées vivrières comme le riz. La part du niébé dans les dépenses alimentaires des ménages est un peu plus élevée en milieu rural qu'en milieu urbain, tel que démontré par les graphiques. Les résultats d'analyse économétrique indiquent qu'une augmentation dans le prix du niébé mène à une réduction des dépenses pour le niébé, c'est-à-dire une diminution de la consommation. Au Sénégal, plusieurs études de recherche sont en cours. Utilisant la base de données collectée auprès de plus de 1000 vendeurs, dont certains qui vendent du niébé et d'autres qui n'en vendent pas, nous sommes en train d'analyser les facteurs qui influencent sur la décision de vendre ou pas du niébé. Nous sommes aussi en train d'examiner si les produits à base de niébé sont davantage une affaire de femmes comme au Mali ou pas. Le questionnaire sur les produits de niébé comportait plusieurs questions sur les variétés. Nous sommes donc en mesure d'analyser si certaines variétés sont davantage utilisées dans la transformation de produits à base de niébé et si oui, quelles sont les caractéristiques appréciées par les vendeurs. Voici un tableau qui résume le nombre de vendeurs par type de produit de niébé et par marché. Parmi les produits, nous y retrouvons les grains de niébé, bien sûr, mais aussi les fourrages, les feuilles fraîches, les feuilles séchées, les gousses, les beignets, la farine et un sandwich de niébé. Certains produits sont similaires entre le Mali et le Sénégal, tandis que d'autres diffèrent. Par exemple, les gousses de niébé ne sont pas couramment vendues dans les marchés au Mali. Celles-ci n'ont même pas été observées lors des enquêtes. Certains produits sont plus fréquemment vendus en milieu urbain, tels que les gousses, les feuilles séchées et le fourrage. Tout comme au Mali, 2 étudiants sénégalais ont participé activement dans les enquêtes terrain de marché. Pour son mémoire de thèse, Djibril focalisera sur la commercialisation du niébé et des produits à base de niébé dans les marchés locaux du Sénégal, avec la prise en compte de la dimension spatiale. Plus précisément, il testera si la gamme de produits varie significativement entre les marchés, si l'intégration entre les marchés est faible et s'il y a une grande variabilité dans les prix entre les marchés. Pour son mémoire de maîtrise, Anna focalisera sur l'offre de grains et de produits à base de niébé sur les marchés locaux au Sénégal. Plus précisément, elle identifiera la gamme de produits offerts sur les marchés urbains et ruraux, cartographiera les différents acteurs et leurs relations ainsi que leurs principales contraintes dans la vente de grains et de produits transformés à base de niébé. L'étude portant sur les changements de préférence des consommateurs et leur niveau de consommation actuel en produits à base de niébé s'appuiera sur les résultats obtenus lors des enquêtes de marché. Nous commencerons la rédaction du protocole de recherche sur les consommateurs lors du dernier trimestre de 2022. Les enquêtes auprès des consommateurs auront lieu l'année prochaine en 2023. Nous allons continuer de discuter la possibilité de collaborer avec le Commissariat à la sécurité alimentaire au Sénégal et l'Institut national de la statistique au Mali. Étant donné notre très bonne expérience avec les systèmes d'information des marchés, nous n'avons pas exclu de collaborer avec eux au cours de l'année à venir. Comme mentionné au début de la présentation, le renforcement des capacités est un objectif de ce projet. Au cours de l'année dernière, le projet a continué de promouvoir le partage des connaissances, le renforcement de la confiance et l'engagement entre les différents partenaires au niveau national, régional et international. Beaucoup de temps et d'énergie est consacré à renforcer les capacités des étudiants. J'aimerais en profiter pour remercier Dr Mamadou Sissoko et Dr Aliou Diagne pour leur appui et efforts continus dans la supervision des étudiants. Tous les partenaires maliens et sénégalais impliqués dans les enquêtes terrain, incluant les étudiants, ont suivi une formation sur l'éthique de recherche. C'est la première fois qu'une telle formation leur était offerte. La formation sur le modèle kaléidoscope a été offerte à tous. Celle-ci a permis aux chercheurs de mieux comprendre les politiques affectant la chaîne de valeur niébé et ceux-ci ont par la suite développé et soumis des propositions de recherche à l'équipe d'administration du laboratoire pour recevoir du financement supplémentaire. La proposition soumise par Dr Aliou Diagne du Sénégal a été retenue. Comme je viens juste de mentionner, Dr Aliou Diagne et son équipe au Sénégal a reçu du financement pour examiner comment mieux intégrer les produits à base de niébé dans les cantines scolaires. Ce projet débutera sous peu. En collaboration avec le projet sur les effets des subventions sur le niébé, dirigé par Dr Milida Smel, nous avons entamé une étude pour éclaircir si le niébé est une culture ou une entreprise de femmes. Cette étude s'appuie sur les données collectées au Mali dans le cadre de ce projet. Malgré les contraintes liées à la pandémie, nous avons continué à interagir avec différents acteurs impliqués dans la chaîne de valeur de niébé. En plus des institutions nationales, nous avons continué nos échanges avec des producteurs, des unités de transformation industrielle et des chercheurs. Une chercheuse malienne nous a même approchés pour qu'on puisse l'aider à publier un livre de recettes sur le niébé. Nous l'avons mis en contact avec l'équipe d'administration du laboratoire. Au Mali, Dr Mamadou Sissoko et ses étudiants ont participé à la foire de jeunes entrepreneurs et ont pu discuter avec une jeune femme entrepreneuse qui transforme le niébé en divers produits, incluant la farine. On voit ici des photos de ces produits dans de jolis emballages modernes. Voici en gros les réalisations faites au cours de l'an dernier. Encore une fois, j'aimerais remercier toute l'équipe pour leur travail au cours de l'année dernière et j'aimerais remercier Dr Mamadou Sissoko pour les photos. Merci.
Thank you, Dr. Terrio, for that very interesting presentation. We have a number of questions for you. If you wouldn't mind joining me on Cam and Mike, there you are. Thank you. So a couple of questions for you, Veronique. First of all, do you plan to characterize some of your findings in a way that they can be incorporated into product profiles for new cowpea varieties?
So the information, it's always, and I think my colleagues, Melinda and Al, you would have better explanation than me. They're definitely the expert when you look at variety. So what we have as information, it's the what it's observed, those characteristics that can be observed on the market. So we're hoping to do some analysis to better understand what the trader are looking for in terms of characteristic that can be observed. Everything that it's not that the trader cannot observe, we cannot control for that. But we're hoping to have a better understanding that why does a trader pick this and not the other one based on those characteristics, to have a better understanding.
Yeah, thank you. I'm thinking that if and when there's a second iteration of the Legume Systems Innovation Lab, that might be a good follow-on for this project.
Yes, and I think a lot of the study, we are going to be looking at the consumer side, but I think it will be a first to look at the vendor. What are the vendor looking for when it's time to make a decision of what to sell?
So this next question is in French, and if you don't mind, I'll translate it into English for our audience. First of all, thank you for your brilliant presentation and the clarity of your pronunciation. This person would like to know what would explain the large difference between the number of markets that were chosen in Mali versus those in Senegal, 26 versus 98.
Okay, great question. So different sampling strategy. So in Senegal, it's nationally representative. And then we also include both trader of cowpea and trader that do not sell any cowpea products. So then we need more observation to make sure that we have a good number of cowpea traders. In Mali, with the situation in Mali, we could not do a nationally representative sample. There are some region up north that we purposely decided not to go. So all the analysis have been done in the southern part of Mali, and different sampling strategy here. We only sampled the cowpea traders. So we didn't have to go and visit as many markets because we were not looking for them to be nationally representative. And we were really focusing exclusively on the cowpea traders.
Thank you, yes. And then is there any regional differences in the cowpea-based products within and across countries, or are they more or less the same?
Good question. So yes, and products are overall some similar, but we are seeing some differences. And one of the example will be the cowpea pods that we don't see at all in the Malian market, whereas they were available across different markets in Senegal. And I'm sure then when we're going to look into more kind of processed cowpea product from like a recipe that people cook at home, then you're going to see more differences across country too.
Great. Well, I see our time is almost up, so I think we'll leave it there. Veronique, thank you very much for your presentation and for patiently answering these questions. Our next presentation is how input subsidies change the legume farming landscape. This project is ongoing and is led by Dr. Melinda Small from Michigan State University. Immediately following Dr. Small's presentation will be a presentation given by project student Natama Maimuna. Natama is a graduate student studying applied economics at University Norbert Zongo in Burkina Faso. The question and answer designation for Dr. Small's presentation is 11, and the student's presentation is 11A. Presenting today for the project will be Dr. Small.
Our project is titled How Input Subsidy Policies Change the Cowpea Farming Landscape in West Africa. Our goal is to better understand the consequences of fertilizer subsidies for cowpea farming communities in West Africa. We have a focus on crop diversity, gender effects, and diet quality. At Michigan State, I'm Belinda Snail. I work with Véronique Thériault and a student, Sibir Ahmad. In Mali, Bourema Kone of the Institut d'Économie Rurale leads the research and supervises 2 students. Amidou Asima has been leading quite a bit of this work. In Burkina Faso, Eugenie Maiga, who is a professor at the Université de Norbert-Zongo, leads this work and supervises a PhD student and 3 MSc students. Last year, I mentioned some results from a paper led by Véronique that was later published in Food Policy. In this paper, we found that the fertilizer subsidy results in a greater concentration of area in crops and target crops and reduced evenness of the area distribution among crops grown by farming households. This is a costly program that clearly favors a non-food crop, which is cotton, and starchy staples, in this case, that would be rice, maize, and to a lesser extent, sorghum and millet, over other nutrient-dense crops such as cowpea or high-value crops. And so this has obvious implications for food and nutrition security. In the second publication last year, we focused a little bit more on farm women. I led this paper. We found that women manage cowpea plots as a primary or a secondary crop, but less frequently than men, although they also work on fields managed by men where cowpea is grown as a second crop. And these fields would be those large fields planted to cereals that are often called the Grand Champs and are collectively worked and supervised by the senior elder male in the household. This is sort of the model in the dryland system. We found that subsidized fertilizer received by the farming household, again, reduces areas and area shares planted with cowpea as an intercrop, and is also negatively associated with women's cowpea harvests and revenues, with the opposite effect on men's revenues. And findings raise questions regarding the design of the subsidy and also gender differentiated effects. The third paper, which was led by Amidou Asima, was published at the end of 2021 in the International Journal of Sustainable Agriculture. Amidou found that total subsidized fertilizer received at the village scale is also negatively associated with the richness of crops grown and incentivizes land allocation from targeted crops away from other crops, such as cowpea. Again, this has agronomic, nutritional, and revenue implications for farming families in these communities. Since he estimated a simultaneous model, he also found that the richer the crop species in a village, the more probable that cowpea is still grown. A paper that Amidou and I worked on together is in review, and in that paper, we ask whether it is the diversity of crops on farms or local market purchases that most contribute to the diet of rural households in Mali. And contrary to what we had seen in the literature, we actually found a very strong effect of crop diversity on farms. And this sort of substantiates that many of these households are still dependent upon what they produce for their food. But we also found a significant effect of purchases in local markets. And so we recommend policy interventions at a regional scale that would enhance the diversity of crops on farms, but also stimulate the trade of diverse food products in local markets. CPR tested the effects of input subsidies on crop diversity with the data set from Burkina Faso, and also found that households receiving the fertilizer subsidy allocate more land to targeted crops than non-targeted crops, less land to cowpea as either a main crop or intercrop, and grow a less rich, less even portfolio of crop species. So these results are robust across data sets and across models, econometric models that we apply. However, he found that the cowpea seed subsidy offsets this bias of the fertilizer subsidy towards targeted crops. Veronique is currently leading an in-depth analysis of the role of women in promoting neglected crops, the case of cowpea in Burkina Faso. And first, in terms of land management and cowpea cultivation, she's found that 75% of households cultivate both collectively and individually managed plots. So this is similar to the structure in the dryland farming areas of Mali, in that a senior male elder in the household supervises collective production on fields that produce the starchy staples for the family, and then allocates individual plots among other household members. And individual plots are exclusively managed by women in a third of the households that are included in the data set. This, by the way, is a nationally representative data set in Burkina Faso. 42% of households cultivate cowpea, and in stark contrast to what we found in Mali, 85% of female plot managers report cowpea as a primary crop, compared to just 5% for male plot managers. And in this case, in Burkina Faso, there is a complex subsidy program that involves not only fertilizer, but seed of a number of crops. So she's looking at the net effects of that program on participating households. And the first thing that she finds has to do with resource control, and that is that the effect of the overall program is fewer individual plots allocated to either male or female household members. And then, sort of as we keep finding in the data sets, regardless of models and regardless of data sets, less land allocated to individual plots managed by women only, less land allocated to cowpea as a primary or a secondary crop, and less land intercropped with cowpea at the household level. The last paper that we're working on in this project is a descriptive paper, and it's being led by Andrea Allen. We're calling it Fact or Folklore. Is cowpea a women's crop or women's enterprise in Mali? And Andrea did a very thorough review of the literature, the gray literature and peer-reviewed literature. And we looked at our large-scale data sets again, seeing that Malian women are less likely to manage cowpea plots than men. They manage smaller cowpea plots, and they sell less, although they earn more from their sales. But we can combine this with the data that were collected under AO12 in a survey that was led by Mamadou Sissoko. And in that survey data, he finds that almost all vendors of processed cowpea products and fresh leaves in local markets are women. That women also retail cowpea grain, but in small quantities, and that men are fewer among cowpea vendors, generally collecting the grain and selling farm. We received some cross-cutting funds to implement 2 nutrition workshops. These were led by Amidou Afsema, in Mali and in Burkina Faso. It was live in Mali and virtual in Burkina Faso. And you see, there were a few students, just, about a dozen in either one of them. This was a hands-on workshop in which Amadou prepared data sets for them, PowerPoint presentations on how to construct and interpret indicators of diet quality, and then spent the afternoons helping them learn how to construct these using Stata. The groups then, the students then broke into small groups to formulate and test hypotheses and report results. Right now, we are conducting a kaleidoscope model case study, and this is being led by Mamadou Sissoko and Amadou Asima, and supported by Buruma Kone and Yanisia Kone in Mali. Most of you are probably aware of the kaleidoscope policy model that was developed by researchers at INFRI and Steve Hadblade, who was at Michigan State University. This is a very interesting qualitative research method that enables you to identify key policy junctures and points of change and circle of influence of stakeholders that have affected these changes. You conduct a series of semi-structured interviews with the stakeholders, and there are some specific tools you use in the analysis. As you can tell from my description, I'm not an expert in this, but the team is doing a great job as far as I can tell. Thank you.
My name is Maimuna Hamana Tama, and I'm a PhD student at Université Norbert Zongo in Burkina Faso. My research is about pathways toward improving the nutritional status of rural people in Burkina Faso, either farm or market. The general fact in developing countries, including Burkina Faso, is that households both buy and produce their food in rural areas. Burkina Faso is a small, landlocked, open economy with high rates of food insecurity. So whether farm production or market development must affect household food security still puzzles economists, because study findings are varied and separating causal effects of each component is difficult. We have two research questions. First, we ask whether crop spices diversity on farms or market participation contributes more to diet quality of farm families in Burkina Faso. Then the second question is about the role of seasonality in changing the effects. In terms of literature, we have two main points. Dietary diversity is a broad measure of nutrition adequacy. We noticed that in our readings. Second thing is that reviews and meta-analysis conclude that the effect of crop diversity on diet diversity is positive but small. So our research goes further by empirically investigating the relative importance of farm and market in improving nutritional status. And the originality of this paper is twofold. First, the construction of 3 indicators to express farm diversity, each based on a specific motivation, and then the test of seasonality. We use cross-sectional data from the World Bank collected in 2014 and which covered the whole country. Also, we use weather data downloaded from NASA site. As I said before, we constructed 3 different indicators to explain on farm diversity. And regarding the market participation variable, we take the natural log of value of crop sales. To express diet diversity, we calculate the household dietary diversity score by season. Let's say that here we have 4 quarters for the year. In terms of explanatory variables, let's say that they are chosen based on the literature. Descriptive statistics points out first the difference, although small, of the household diet diversity score during the year. Also, it shows the different distributions of the 3 indicators, although both express on-farm diversity, they are different by definition, and each of them, as I said before, is constructed based on specific motivation. So, for example, the Convening expresses the richness, while Simpson expresses richness and regularity, and cereal area share is the percentage of land area allocated to cereals. Also, descriptive statistics show here that there is a high variability of the distribution of the variable for market participation. So, we use the conditional mixed process model developed by Rudman in 2011. This approach addresses endogeneity in a simultaneous equation framework. The model is regressed with full information maximum likelihood. And we use a set of explanatory variables related to household, market, plot, and weather characteristics. In terms of results, let's say first that market participation is always positive and smaller, regardless of the specification. Second, we notice that the effects of Convening and Simpson indexes are both positive, while the effect of cereal area share is negative. Simpson has actually greater effect among the 3, showing that the count of crops is not uniquely associated to nutrition, but the size allocated for each crop plays an essential role. Third thing is that the negative relation between the cereal area share and the diet diversity can be explained as a confirmation that household consume cereals and the agriculture is extensive in Burkina Faso. So we sum up our findings into 2 points. First, on-farm diversity has a greater impact on diet diversity than market participation. And this is contrary to findings in much of the literature, but it's consistent with findings for Mali. Second thing is about seasonality, which we notice eliminates one of the 2 effects at certain periods during the year. In terms of conclusion, both farm and market are associated to diet diversity, but the farm effects are greater. The results show clearly that farming households continue to rely heavily on their production for food. Our findings are in favor of interventions tending to cautiously improve on-farm diversity, but our findings are not encouraging expanding area allocated to cereals. So the interventions are, for instance, seed supply, because market-based incentives for diverse crops at the regional scale could strengthen both farm and market pathways. Moreover, findings suggest to take seasonality into account when implementing the policies. We reached the end of our presentation. Thank you for your attention.
Thank you very much, Dr. Small and Namata, for your presentations. I would like to invite Dr. Small to join me on cam and mic so that we can address some questions to you. And Namata is available to answer questions in writing. She won't be appearing here in the live question and answer session, but if you have questions for her, just be sure to mark them as 11A in the question section, and she will answer them in writing. So Dr. Small, first question, do you have any policy recommendations regarding fertilizer subsidies based on your findings? How should subsidies be structured to avoid discriminating against women?
Well, there are other examples of fertilizer subsidy designs throughout Africa that might be at least studied in the context of both Burkina Faso and Mali. In the case of Burkina Faso, the policy does seem more complex and to include more crops, but in other parts of sub-Saharan Africa, subsidies have been specifically targeted to vulnerable people. So to make a decision on whether the design, the current design is better than an alternative design, you really need more information. And I think that sort of research is important and needs to be conducted within these countries. So I personally would not go so far as to make a specific recommendation of the form of the policy, but I would recommend that the design be revisited, constantly reviewed, with an eye to not only its bias towards starchy staples, but its bias towards particularly well-endowed farmers.
Understood. And another question is, are seed subsidies targeted to newer improved varieties to encourage varietal turnover? What is the macroeconomic benefit for a country to offer this? And how does this interact with and support the informal seed system?
In the case, we did not study seed subsidies in Mali. There are some, I believe, some seed subsidies on improved starchy staple crops, improved varieties of starchy staple crops, but they're not part of our data set. In the case of Burkina Faso, we're still looking at this in greater detail. It could be that in the seed subsidy programs in Burkina Faso, some of the improved varieties are included. We're on the lookout for that for cowpea.
Understood. The next question is, well, it starts with a commendation. They commend you for ensuring that project results are shared to the wider research and policy community through their active engagement in scholarly work production in peer-reviewed journals. And then the question is the last paper going to be peer-reviewed too, or will it be published as a popular article?
I think we're thinking of submitting it to a journal that focuses on gender research, but the kaleidoscope policy model case study that we're conducting, we really want to use as an opportunity to engage with stakeholders. So we have an extension on our project. We've actually completed our project time, but we have a no-cost extension. And during this extension, we're going to be focusing on stakeholder fora in the two countries. The work by the students is not yet complete either. And I believe that, as you've heard Maimuna's presentation, there will be some exciting research findings. And we're hoping that in Burkina, that's what I think we would like to extend and perhaps do an article in the local newspaper.
Understood.
A popular article.
Great, yeah. One final question, based on your research, oh, excuse me, this one I think has already been asked, or at least a form of it's been asked. Let's try another one. How do you find the process of advising students at other institutions? Is that something that's been easy, or has it been difficult?
Sorry, we don't, our leads in our study countries advise our students. We have some, it's always nice for us to be able to engage with them, but the direct advisory responsibility is always Bore Makone or Ujani. So I'm not sure if I understood that question well.
Okay. Well, how about this one? Are you already seeing impacts from high fertilizer costs and scarcity? And if that did not occur during your project, what do you think the effect will be on these two countries?
That's right. That did not happen at the time that our data were collected and that we were conducting this analysis, if you're talking about this season. I have no idea how that might be and maybe impacting these two countries, but I would think it would be a very serious impact. It's going to be a serious impact in the US, for example.
Yeah, and I imagine that what governments are able to do is going to be greatly reduced just based on the cost. So, yeah. So thank you, Dr. Small, for those answers. We would like to now move to a short presentation of our RT video. And let me just make sure I can see which one it is here. This one is on our efforts in the area of youth. Our next research presentation is on the Cowpea Atlas Phase 1, Mapping Cowpea Data Sources and Gaps in West Africa. This project is ongoing and is led by Dr. Usman Coulibaly from CERAS ISRA. The question and answer designation for Dr. Coulibaly's presentation is 12. And presenting today for us is Dr. Coulibaly.
Cowpea Atlas projects. This is the title of our core project. So it's divided into two phases. The phase 1 is mapping cowpea data sources and gaps in West Africa with 4 countries: Benin, Niger, Nigeria, and Senegal. Cowpea, Vigna unguiculata, is an important legume in arid and semi-arid zone of Africa. It has economic, nutritional, and agronomic benefits with cheap source of protein, minerals, and vitamins. And also, it has a high nitrogen fixation for poor soils. And now, along the cowpea value chains, there are many cowpea-based food products, including the infant food, the mixed cereal, cowpea flours, and other diversified food. Along the cowpea value chains, there are women, men, and youth, all engaged in the various segments of the cowpea value chains. But there are strong social, cultural, and economic factors which constrain cowpea production, cowpea storage, and marketing. And this is mainly due to the lack of information and reliable data along the value chains. So the main objective of this project is to make information accessible to stakeholders through an online platforms. And this will be done in phase 2, while in phase 1, we are doing the mapping of cowpea data sources and gaps along the cowpea value chains. So specifically, in phase 1, we are identifying cowpea sources and then look at various segments of the cowpea value chains in target countries to see what type of data are available, and then to assess these data sources and also show the gaps so which can be filled later. And as I mentioned before, the project is implemented in 4 countries: Benin, Niger, Nigeria, and Senegal. The host and solutions. So we are based at CIRAS, ISRA in Chess, which is a regional research center. And there are 4 main institutions where the project is located. In Benin, it's located at GBIOS, a genetic resource lab based at the University of Abomey-Calavi in Benin, Cotonou. The Institute of Agricultural Research based in Zaria, northern Nigeria. And the Institut National de la Recherche Agronomique of Niger based in Niamey, in Iran. And the Bureau d'Analyse Macroeconomique BAM based at ISRA in Dakar. But we have a wide range of partners and stakeholders which provide us with data, information, and when we are developing collaborative ties. So USAID and USDA for the food security components in Washington, with CGR systems with big data, the RTA, IFPRI for policy research. RTA is mainly for agronomy and genetical resources, and PABRA CIAT, so based in Nairobi, which can provide us also some guideline for making an atlas, because we made one out of beans. And we have also a lot of information from the Future Innovation Labs and collaborative research support programs. So there is a good source of data information. The World Bank, African Development Bank, food security components, FAO, World Food Programs, Fuse Nets. So for food security and cross-border trade data available. So we are working with them. Coraf also, which has experience in doing the platforms. So we are going to work with them to make sure we will establish our platforms on cowpea. We have sales agreements with Niger. So we have a lot of information also on climate change, which can be useful for this project. And there is the market information systems available at different ministries within the countries. And of course, in each country, there are some statistical data for national food security or the Ministry of Agriculture and Trade, so which can provide us with a lot of information, and which can partner also, like for the sustainability of the data collection process. For the outcome and the impact, so the intermediate results, we should have copy database containing information on copy value chains, which has to be available in targeted countries. And for the outcome, the stakeholders involved in copy value chains can make use of this database and also the gaps to plan further actions aiming at boosting copy value chains. And also, we are assessing the copy data sources and gaps along the copy value chains in target countries. The overall impact is to increase the copy production, the copy consumption, and the copy marketing. So when we look at the project's activities, so there are 3 sets of sequences. One, in phase 1, identification of data sources and gaps, what we are doing now. And moving toward the phase 2, we are strengthening also the data collection systems to make sure that we leave a system which can really repeat itself and sustainably after the project is ended. And the second phase is mainly to consolidate this data and make a virtual copy atlas platform, which can be fed by different stakeholders and in a very organized manner to get information and data for the economic purposes. So when we look at the type of data collected, so we are collecting all the data along all the segments from research, so varieties, germplasm, seeds, inputs, producers, farmer, markets, processors. So we have a group of actors, and then we have really some disaggregation also levels, like the type of seed or type of markets. So when we look at like the setting of activities and the flow of activities, so the advisory national and regional panels, we are setting them now, and they will be finished during this period, which is really the third part of the project. The green is the activities which are already completed. The blue are activities which are ongoing, and the red, which are activities planned. So the identification of data sources and availability along the segments are continuous activities from Q1 to Q4. And the data gaps in the copy value chains are planned end of Q3 and the whole Q4. And the data aggregations also is ongoing, but it will be finished in Q4. And the data screening for validity and reporting is also an ongoing process. So from Q1 to the Q3 and 4. Now, when we look at the advisory national and regional panels, so in Benin, this has been installed already and it's working. And we have 11 members, 9 men and 2 women. In Nigeria, it has been also installed. The first meeting has been held in February, and it has 15 members, 13 men, 2 women. In Niger, it's ongoing. We have 16 members identified already, 11 men and 5 women. In Senegal also, it's ongoing. And then there are 11 members identified, 8 men and 3 women. So for the data collection, the secondary data collection, the data are collected from the platforms, reports, published articles, and copy. And also we are going to rely on panel members to help identify more data sources and to sustain the data collection system after the lifespan of these projects. Way forward, so we are now preparing to start field data collection, primary data. This is in March, ongoing now. And we are checking also the data collected for reliability and accuracy. And we are going to share the data collected via Google Data Studio dashboard. Thank you for your attention.
Thank you, Dr. Coulibaly, for that very interesting presentation. We have a couple of questions for you, so I would ask you to please join me on cam so that you can address these questions. The first one is, why was Burkina Faso as the third cowpea producing country not considered for this project?
Yeah, well, I think this question always comes. Yes, we have to make a choice. And given the budgets, we say, well, let's see are consuming cowpea. So Nigeria was chosen. Then Niger was the second one to be chosen. And then Benin has been chosen because the capacity of GBIOS to do data monitoring and evaluation, I think this was very important. And then Senegal, of course, Senegal has been chosen because of CIRAS, but because of all the linkages and exchanges between Senegal, Mali, Senegal, Mauritania, Senegal, Guinea-Bissau. So this was really a criterion. But yes, Burkina Faso is really a big producer. If there is anything else, I think I will choose Burkina Faso. But now we couldn't. The Quora has always asked me, why not Burkina Faso? A good question, legitimate question, but you know, resource constraints.
Mm-hmm. Yeah, thank you, Dr. Coulibaly. And if I may add, I think, and you can correct me if I am wrong, but the intention is that this was going to be a pilot, and that once this pilot is completed, then other data sources would be added to the platform over time. Is that correct?
Yes, this is correct. So
yeah,
this is a pilot.
Yeah.
Yeah, exactly, including Burkina Faso.
Correct, yes. Okay,
also
thank you.
Other crops. Yeah.
Mm-hmm. Yeah, thank you for clarifying. And then the next question is, are the National Agricultural Research Systems part of the partners? And will you be reaching out to them?
Yeah, if you look at the countries, and all the countries, I think the host national agricultural system and national research systems. So all the universities, like in Benin is a university, but we are doing also research. But when we look at the stakeholder composition, we didn't present it. There is INERAB, the National Ag Research System, which is part of the panel system. And this is very important because after this project, so the panels will be the key for the sustainability of the data collection, the data analysis beyond the lifespan of this project. Yes, in each country, you know, like the national systems are included.
Yeah, thank you. And then how is this project linked and or aligned with other Atlas-related initiatives, especially related to crops, agri-farming, etc., in sub-Saharan Africa and other countries? In other words, how are you relating to other platforms that are out there? Have you talked to them? Are you integrated with them? What is the engagement that you have with some of the existing platforms?
Yeah, this is a really good question. When we look at platforms, and Quora has now 2 to 3 platforms. So there is a platform for inputs, data, and input prices, and we are working. So we had a meeting with them to see how we can link up with them. So for guidance, and see if even, we can locate our platform inside the Quora because of their experience. And we are going to have a meeting with them in April. So to make sure we have really a kind of workable schedule of activities and contribution from them. And the second platform is the common bean platforms from SIAT. And I wrote to Jean-Claude, so from SIAT, to seek for some collaboration. And I got even, the atlas from the bean copy, the bean atlas. So we are going really to work with them. So to see how we can use the experience and even the data set, the way we put the data together. So to make the platform, how we can really build on this also for the copy one. Yes.
Fantastic. That's great news. And we look forward to phase 2 of this project. At this time, thank you, Dr. Coulibaly. Thank you so much. At this time, I would like to welcome Dr. Barry Pittendry to provide a short presentation on the Legume Systems Innovation Lab Associate Award, Feed the Future Scientific Animations Without Borders, Responsive, Adaptive, Participatory Information Dissemination Scaling Program, fondly known as SAWBO Rapid. This will be the final research presentation of the day, and the question and answer designation for Dr. Pittendry's presentation is the number 13. That is the number 13. Over to you, Barry.
Great, thank you very much, John. And David, if you can just pull up the slide presentation, that'd be great. So welcome everybody. Appreciate the opportunity to speak today on the SAWBO Rapid program. Next slide, please. The SAWBO Rapid program was an associate award that was provided to the Legumes System Innovation Lab at the very beginning of the COVID-19 crisis, and looking towards two levels of issues. One is during the COVID crisis, how could we develop educational content that could help people deal with resilience within the context of this crisis, around everything from food storage to how to properly handle themselves in marketplaces to minimize exposure to COVID. But more broadly speaking, and I would like to put this in sort of a broader conceptual framework, one of the challenges that we have is how do we move things rapidly through a product life cycle to get things out the door into people's hands as rapidly as possible. This program really, it was exploring this space around this particular issue of how do we go from innovations where we have the evidence that those innovations can help people to get them into their hands as rapidly as possible so that we can begin to have impact. Next slide, please. So just thinking towards the fact that all of your programs are producing great innovations. Some of these innovations, once they are out in people's hands, they're going to have tremendous impact for generations to come. And I would argue that innovations really fall in probably 3 different categories, but for simplicity's sake, I'm going to talk about 2. Of course, policy is incredibly important in driving innovation, but I won't talk about that within the context of this presentation today. But for simplicity's sake, let's think about innovations falling under really 2 categories: educational interventions or widgets or products. And those may be physical or tangible things, an improved seed, a new device that's more effective in helping us move forward a particular value chain that we're working in. As we go through these systems, there's often an investment and basic research that brings us forward to this valley where we get those papers out into journals, and those innovations are there publicly available, but may not reach the final end users. Then continued investments may take those from basic research developments to research for development innovations, where we have all that necessary evidence to show, yes, this innovation will help people, and it will help them in multiple, as we look at multiple parameters. Is it gender sensitive? Does it help youth? Does it fit into a particular type of ecosystem where it can be useful and help people? The challenge often has been is how do we get these innovations out to people's hands and how do we do it at scale? Next slide, please. So the focus of the Scientific Animation Without Research program, for those of that you've a biological background, is to be a catalyst to make it much more easy to get innovations from this step out into people's hands. In order to do that, our goal is to make things far more accessible and dramatically drive down the costs of taking innovations from the stage where we have the evidence that is going to be useful to the stage of getting it into people's hands. Next slide, please. So there's a lot of great concepts out there that took a long time to go from inception to having very pragmatic impacts in our lives. Think of Alexander Graham Bell, probably well over 100 years ago now, and then you're probably all looking at your smartphones right now, or you've got them at least beside you. There was a big shift from the concept to making it inexpensive and then highly accessible. And as I have a background in molecular biology, I always like to use molecular biology examples. So here we have Watson and Crick on the left-hand side. This is the early 1950s when they published what we now is a foundation of what we know about how genetics contributes to what we know as life. But how did that move into the practical space where it's impacting every one of us? Well, we've all had COVID tests recently, or at least within the last year, or many of us have. And all these technologies became inexpensive and highly accessible. So in the early 2000s, the real shift came when automation occurred, when we could drive down dramatically the costs of this technology and dramatically increase what we could get out the other end. So driving down costs, increasing accessibility. Next slide, please. So the take home is how can we get better bang for our buck? How can we get innovations out rapidly to people, have impact, and get it out to more people for less money? So this is, I think, in my opinion, the long-term goal is how we're going to get more things over that hump out into the field is through these type of strategies of getting better bang for our buck. Next slide, please. So just to give this some context, the Scientific Animation Without Borders program is about a 10-year-old program where we take global experts, work with global experts. We take ideas and concepts, our life-improving concepts, we put them in 3-dimensional, 2-dimensional animations, put them into numerous languages. It goes into highly accessible libraries that can be disseminated in numerous platforms, handed off to other groups that can scale. Our goal is to create, they scale, and then to study how those things are scaled. And there's over a decade worth of work on showing that people will learn by this mechanism. They have learning gains. They will. There's evidence of adoption with some of this content and innovation with it as well too. And where do you need to interject it into given systems in order to have impact? So all that research was funded by USAID and previous innovation labs for about a decade before this, leading up to this event where we can begin to ask the question of how can we scale this to large numbers of people and then look to drive down costs and make it as effective as possible. Next slide, please. So we create content. The content we can continually adapt it. We can add new languages. We can add new cultural variants. We can reach out to the groups of people that we work with and continually add that content to the system. The system grows, and as more content gets added and more languages get added, it becomes cheaper and cheaper and cheaper. So we can drive down costs per unit of valuable information as the system grows. But it's also a deployment system as well. Next slide, please. So Scientific Animation Without Borders, it's a research and scaling program with a global reach. We content over the past 10 years, about 100 different 20 topic areas. We have evidence of content at least touching people in at least 130 different countries. And between known use on TV stations and YouTube, well over 150 million people touched. The recent SABO rapid funding mechanism has allowed us to ask some bigger picture questions of how can we scale and how can we drive down costs. The this content can then be passed to other groups that can scale it. Recently, prior to the Sabo Rapid program, we engaged with beginning to ask questions using research machine learning tools. So next slide, please. And this will put in context where we're going with our existing data sets that have emerged from this. So what we, in a previous project where over 134,000 farmers were trained in Bangladesh using these animations, we collected collaborative groups that collected large amounts of data. And from those data, we were able to use machine learning tools to develop models to ask, what is the best place, time, and environment to optimize gender balance? So by using these large data sets, we could actually ask questions of, what is the best circumstance to optimize gender balance in training sessions? So from the extension efforts, we can do the research to understand how to do better extension efforts and how to deploy to more people while maintaining, for this case, gender balance. But these type of shifts have allowed us to ask bigger sets of questions. Next slide, please. So the Sabo Rapid program existed in 4 different countries: Ghana, Nigeria, Kenya, and Bangladesh. Next slide, please. And what we did is a series of different questions. First, we had to ask, what were the problems that were going on in these countries? What were the interjection points where we could bring valuable, scientifically validated information into a system to help people with a challenge we're facing during COVID-19? So we did these essentially market assessments to understand where there were gaps in knowledge that could be filled in order to have a positive outcome for people. Next slide, please. We created the content with local partners, with the missions, with our USAID partners in Washington, and with partners that they engage us with, including Johns Hopkins University, around issues with COVID. Content, once created, was placed in numerous different languages and made available for other groups that could deploy by WhatsApp pathways, could be broadcast on TV stations, could be used in many, many different mechanisms. Passed off to partner groups and then work with them to deploy and understand what was successful in deployment. Next slide, please. So for example, in Kenya, a local group was able to set up a countrywide WhatsApp network where they had well over 250, close to 500 now, because the system's still on the go and expanding, extension agents across the country that they would share videos with on a regular basis. And with this content being in dozens of languages for Kenya, they could pick and choose when and where to use it and when and where to deploy it with their neighbors throughout the country. However, other groups put it on television stations. We also pushed these through large YouTube advertising campaigns that went out to multiple millions of people in short periods of time. Next slide, please. We were able to collect data out the back end. This is just a broad slide that deals with the data collection system to understand when and where content was being used. So we have very large, I'd say, unstructured data sets, which is what's make them very useful for machine learning type of approaches. Next slide, please. And where we stood with this program, it was we're able to deploy content out to multiple millions of people and actually be able to observe what watch time they had. We're able to study when and where this content was being used. We did 3 large-scale studies, and this is work that was done by, driven by John Medendorf. And I also like to mention that John Medendorf, Julia Bell Bravo, and myself were the 3 PIs on this. John put in a very extensive penetration study network to look at penetration and how much uptake occurred with this. And we're now at the point where we're beginning to look at the cost per person touched and using these type of models to understand how to drive down cost. We're able to develop multiple scalable approaches using YouTube ad pushes, WhatsApp networks, TV stations, and we were able to develop 10 major content topic areas and place it in numerous languages, with one in over 100 languages for the 4 countries we're working in. And what has emerged from this is ability for us to look at exact costs per person touched and looking towards a relationship between individuals that observe this and what percentage we're adopting and cost per adoption. At the tail end of this project, we're able to demonstrate, really we're able to drive this down to pennies per person touched and into the dimes or low dollars of people around issues associated with adoption. Next slide, please. So thank you. I'd just like to thank USAID for all their efforts on this. And there was a rather large team of people involved in this. But I'd like to thank John Medendorf and Janet and Julia and the rest of the team members for all their efforts to make this a success in the 19 months that we worked on this program. Thank you very much.
Thank you, Dr. Pittendreen. If you don't mind just staying on camera there, we'll ask a few questions. First of all, how do you reach people who do not happen to have a smartphone or a computer or access to the grid?
That's actually a really great question. So early on, we did work with Northwestern University. This is years ago and published work on this of a two-step process. So you will have a particular deployer that can receive the information, an individual that would be the extension agent. They can receive that information into the village or wherever they're at. And then that can be used, their smartphone or cell phone can be used to basically as a mini viewing station for people that don't have that. And this is what we found with this deployment through a WhatsApp network is many of the end users did not have access to this technology. All you needed is somebody in a village or that was going to go into the village that was at the other end of that WhatsApp network could receive that information and then use it in whatever extension efforts they were doing when they would go out to the village. So the big issue is getting it to the people that will do the final deployment and integrate it into their educational programs. Great question.
Yeah, and the next one also I think is a great question. Are these videos also produced in local languages in order to reach more people?
Absolutely. So all content has been put into numerous languages. So we have content in over 260 different languages for the SABO program. In Kenya, not only were we able to achieve, I think it was well over, for at least one of the videos, well over 50% of the languages for Kenya, but in one language, we're able to get at least a half a dozen to a dozen different dialects of a given language. So then the system that we have online, people can easily choose the language and actually in some cases accent variant they want, download it and run with it. So this is one of our, I would argue, one of our great strengths is our ability to place things into numerous languages. And this was candidly, from my perspective, what was very exciting is a previous USAID investment, which was how to store beans in jerry cans. We were able to get this in close to 100 languages across these 4 countries. And that includes dialect variants as well. So no, absolutely. And once the content's created, it's really easy to get into more languages. And it becomes incredibly inexpensive to place it in more and more languages as we move forward.
And another and final question is there a cost to deployers, an extension to share or use the animation? Is there any cost for using the videos?
Absolutely not. So our mechanism is once content is created, it's freely available for anybody who wants to use it for educational purposes. And there's no cost to an end user in that sense. So our approach is to make that content, make it freely available. And I'll argue very vehemently that we tap into the academic model in this. So as we think about when we publish papers, yes, it takes resources to publish papers, but when we're working collaboratively with other people, there's a lot of goodwill that goes into making sure publications happen. So we reach out to global experts who contribute their time. We reach out to linguistic experts that contribute their time because at the other end, they know that people will have free access to this information.
Thank you. Thank you, Dr. Pittendreen. That concludes our research session for the Feed the Future Innovation Lab for Legume Systems Research Global Virtual Convening 2022. At this time, I'd like to turn the meeting over to Doreen for some final housekeeping notes. Doreen, over to you.
Thank you, John. As we near the conclusion of today's public forum of the Legume Systems Innovation Lab's Global Virtual Convening 2022, I'd like to review a few reminders. All of the pre-recorded research presentations will be available for reviewing soon. These will be able to be accessed in the Hoover platform, on our Legume Lab website, or directly on our YouTube channel. We will alert you via email when these recordings are available. Also, continue to check out the community feature in Hoover, where you can engage with other attendees and find additional resource materials. Our presenters will also work to provide written responses to all questions addressed during their presentation. Look for their answers in their session Q&A feature. Also, feel free to contact them via the Hoover messaging feature. The Hoover platform will remain accessible to all attendees. A quick reminder for our project personnel, we look forward to meeting with you tomorrow during our project strategy session, which begins at 10 a.m. Eastern Daylight Time. At this time, I would like to welcome back Dr. Barry Pindry for convening closing remarks.
Thanks, Doreen. As we close the public forum on the Legume Systems Innovation Lab Global Virtual Convening of 2022, I would like to thank everyone that made this event and this research possible. USAID, we can not thank USAID enough for all the resources, time and effort, and collaborative interactions that we've had with them. I think it's important to note the incredible amount of time that's spent by USAID staff to work with us on this, and we appreciate every minute you spend with us. It's a wonderful collaborative interaction. Michigan State University for their ongoing and many years of support for this program and previous programs. Purdue University, our advisory boards, the Link and the TMAC. Both the Link and the TMAC put a lot of time and effort into helping us move this program forward. I'd like to thank the lab management entity for all the work that they've done over the past several years, and of course for this event. And all of our research project teams and collaborators, you're doing wonderful work that will have impact in people's lives for years to come. So thank you for your dedication to this program. The last 2 days have been full of exciting news and updates on innovative lab, legume lab research to challenge both global hunger and help make the world a better place. Legumes offer high protein and nutrients at a low cost, plant-based alternatives to high carbon footprint animal products, and are kind to the soils through their nitrogen fixing properties. Legumes are well positioned to play a major role in feeding and caring for our world. I've been inspired by the young researchers who shared their visions with us over the last 2 days, and I'm confident that they will carry on with a legacy of dedication to science and their communities. So as I close this year's convening, I look forward to the next. As we opened our first program, a global convening in Senegal with in-person meetings, our goal is to make our last program global convening in 2023 face-to-face in Southern Africa. Stay tuned for more details, and thank you for joining us yesterday and today. Have a good day. Thank you very much.